Hello and good morning po sa
Good
inyong
morning
lahat.
po.
Thanks for joining Saturday. Today is a Tuesday. It's not a Saturday, it's a Tuesday. And we're very happy po to have another very interesting talk po ngayong araw na to. And our title po is Hunting the COVID-19 Variants. Paano po natin nalalaman na may variants na po sa Pilipinas? Paano po natin nalaman na meron na pong bagong P3 Philippines variant ng COVID-19? And who are the scientists who discovered this very important and groundbreaking discovery po. So today, malalaman po natin yan with our special guest. So good morning everyone to this Tuesday. I'm Dr. Ana York Bondok. I am an internist, a pulmonary specialist, and a critical care specialist. Our program po is called Libreng Konsulta. And sinusubukan po talaga namin, and it's our goal to bring science and health closer po sa lahat po ng tao. So, meron po kami YouTube channel. It's called Dr. Ana York Bondok. Please follow us po on YouTube. And sa mga may tanong po today about COVID variants, kung paano po sila na-discover, kung paano po meron ng P3 or Philippines variant, ilagay lang po sa baba. If you are very interested, please share this, lalo na po yung mga doktor and future young scientists. So let's start. Our title po is Hunting the COVID Variants, or how we discovered the P3 variant dito po sa Pilipinas. We are very honored to have as our special guest. She is the Executive Director of the Philippine Genome Center, which is the agency po of the University of the Philippines and of the government po that discovers and sequences COVID-19 variants in the Philippines. They are the discoverer of the P3 Philippines variant. We are so happy to have one of the most eminent scientists po sa Pilipinas and a molecular biologist. We're joined by Dr. Cynthia Palmes Saloma. Thanks, Ma'am Cynthia, and Professor for agreeing po to join us today.
Oh, thank you, Dr. Ana. It's a privilege for me to talk to you today and also to our audience. And hopefully, I can explain in simple terms and in simple words the work that we are doing as part of the IITF, a technical working group on COVID variants, as well as, of course, the work that we do at the Philippine Genome Center.
Yes, ma'am. Na-intriga po talaga ako dyan sa Philippine Genome Center. I'm already a practicing physician, but it's only now po na nalaman ko, though I had heard po of the test kit na na-develop. So, relax lang po tayo para maintindihan po natin lahat. At para maintindihan ko rin, even though I'm a doctor, I still find it difficult. So, ma'am, we're honored to have you today. So, let's start po. Tell us po, kasi ako po, nalaman ko lang na may gene sequencing na po sa Pilipinas nung labasan po itong mga variants na ito. And that eventually we found out that they were a big problem po because naiba po sila from the regular COVID. So, can you tell us po, sino po ang nagsisequence and what is sequencing and who does the sequencing po of the variants or the COVIDs dito po sa Pilipinas? And ano po yung inyo pong Philippine Genome Center?
Dr. Ana, the Philippine Genome Center was started in 2009 by the University of the Philippines Board of Regents. So we started out as virtual facility. Wala kami masyadong staff in the beginning. But what really started the story of the Philippine Genome Center was yung ating pag-host ng HUGO Convention in Asia Pacific, the Human Genome Organization Conference in Cebu. At ang ating organizer niyan is yung current Chancellor of the University of the Philippines, Manila, si Dr. Carmen Zita Padilla. So doon sa Cebu, there were a lot of scientists with very excellent presentations. At nandoon din po si President Merlinda Roman. Tapos tinanong niya si Dr. Padilla na meron ba tayong ganito sa Pilipinas? At sinabi nga ni Dr. Padilla na wala pa po. So ang binigay sa amin, we were given a mission, 4 kaming mga babae, so na mag-isip kung ano yung pwede nating ma-craft na dapat sa Pilipinas kasi wala talaga tayo noong sequencing facility na pwede tayong pumunta kasi dati palagi natin pinapadala pa yung ating mga sequences abroad and it really created a lot of delay at saka syempre mahirap mag-transfer of funds abroad. So Dr. Ana, from there, kaming apat, yung Vice President ng UP noon, si Dr. Amelia Guevara, tapos si Dr. Carmencita Padilla, at saka si Dr. Gisela Concepcion, at ako, apat kami, we went around Southeast Asia at saka sa US na rin, talking to the Filipino diaspora and asking many of the scientists running the Genome Center labs in Hong Kong, in Taiwan, and also, of course, in Baylor, in New York, and in the Broad Institute in Boston. So we were trying to craft a genome center that would be fit for the Philippines. Of course, during that time, wala pa siyang pangalang Philippine Genome Center. So that was in April and March, April. And then by July, naipasa na ng University of the Philippines Board of Regents yung creation of the Philippine Genome Center. Pero during that time, wala pa tayong laboratory at kung ano-ano pa. So it was really a virtual facility. Then I was tasked to craft the proposal na magsimula nitong ating DNA sequencing core facility. So I started it through the help of the DOST-PCR. So kumbaga, yung genomic sequencing facility natin was really a flagship project of the DOST together with the University of the Philippines. So, it was really a triumph when sa Secretary Montejo at that time and the other officials of the university and of the DOST and also ngayon ang current undersecretary natin si Dr. Rowena Guevara, when we all were together and doon din si Dr. Roman na i-open na natin yung ating Philippine Genome Center. Noon sa MBB pa lang ito kasi magkatabi lang naman kami ng PGC at saka of course the effort was really also spearheaded by then President Pascual of the UP system. So nagkaroon tayo ng ganyan. So what was the basis bakit tayo nagkaroon ng isang Philippine Genome Center? Number one, sinabi ko sa inyo na talagang ang mga researchers hirap na hirap magpadala ng sequences nila. So for example, kami, molecular biologists kami. We do a lot of genetic engineering. Kailangan namin palagi i-check whether our construct is correct, whether the sequences we have are correct, and whether, syempre, yung pagdikit-dikit namin ng mga genes, at the same time, gamitin mo rin siya sa testing, Wala. There was a time pala we were going to San Lazaro Sakil lab for a while, just to have sequencing, pero nahirapan talaga kami. So, we decided to have it here in the Philippines. And it was also a little bit difficult, Dr. Ana, kasi the technology was moving very fast. And it was also a bit risky to buy expensive equipment at a time na medyo yung mga laboratories hindi pa masyadong handa. So, may first phase tayo and that was really the help of the DOST. Tapos, meron naman tayong second phase and this is really the powerhouse equipment that we have now. And this has been facilitated by by the Ched Picari. So, Ched Picari granted the University of the Philippines system something like 300 million pesos for us to be able to buy all these equipment in anticipation also na when the borders are closed and we actually anticipated a pandemic, because of the experience in SARS in Hong Kong na pag wala ka, the borders are closed, then you have nowhere to send your samples to. So kasama yan sa ating justification on why we need to have a sequencing facility in our country. So we opened the DNA sequencing core facility in 2012. Doon pa yon sa bagong building ng MBB, sa National Science Complex, until such time na we moved here to our permanent home, the new Philippine Genome Center building in 2017. So, Dr. Ana, would you like me to share a screen? Gusto ko lang ipakita sa kanila yung ating mga picture at anong makikita sa Philippine Genome Center inside of it, Kasi it's really a state-of-the-art equipment and we're really, really thankful to the past executive directors of the PGC, si Dr. Amelia Guevara, si Dr. Menchit, si Dr. Barles Aguda, and of course, I'm now the current executive director for all their efforts. So, i-share ko, doktora share screen.
Ang ganda-ganda ng kwento, lalo na it is International Women's Month. 4 na babae
Correct.
pala.
Oo, 4 na babae yan.
At po, 4 na Maria. Opo.
4 na Maria, opo. Tapos, it's
for women saving the Philippines, ma'am. Ganda ng kwento.
At saka, when we started, during the opening ceremony, it was really a touching moment, Dr. Ana, kasi parang, siyempre, maraming mga plano-plano din never materialize. Diba? Daming programs, ginagawa, wala naman nangyayari. Pero yon, that was a physical entity na pwede na tayong mag-sequence dito mismo sa Pilipinas. At nag-expand pa siya
Wow.
noong mga 2015 kasi si Doktora Eva talaga champion dito. Doktora Eva Cotionco de la Paz,
Opo,
ang
kilala po.
daming tears and sweat ang binuhos ni Doktora Eva para magkaroon tayo ng Ched-Picari laboratory. Ito naman yung kumbaga nag-upgrade tayo into a more much state-of-the-art equipment, and these are the equipment we are using now. So, Dr.
Sige
Ana, di
ma'am,
ba, oo,
minsan
pakita ko sa inyo, no?
opo.
Oo, kasi
Yes, we are
pala,
so curious kasi we don't
oo,
know much. We would like to learn. Opo.
oo. Ano nga yung aking gagawin para ko ma-share screen, StreamYard,
Sige
entire
ma'am.
screen,
Opo. Miss
okay,
Kat,
yan.
please
And
help,
then
ano.
allow, I think I can figure it out. Is it working now?
Yan na po.
Oo, so yan. So I'd like to introduce the audience, no? So ito po yung ang facade ng ating building. So this was also so this is a state-of-the-art facility kasi ready tayo sa mga pressurization ng lab. So nung kailangan nating mag-expand ng COVID testing, we converted the entire floor nitong third floor natin. So this is itong sa right side in the picture, in the image, ito po yung ating laboratory wing, no? So first floor, dito sa first floor, ito yung ating unang set of equipment. Tapos the second floor, ito naman yung ating clinical genomics at saka third floor. So na-mention mo sa iyo, Dr. Ana, na we were actually planning to launch yung aming services sa cancer testing dito sa clinical genomics facility ng PGC na medyo na-postpone a little bit dahil nga dito sa COVID testing. So this is the facade of the Philippine Genome Center. This is our permanent home. We moved here in 2017. Pero bago yan, katabi lang, ang likod nitong building na to, this is called the MBB building. So ito din yung yung ating source ng talent, no, ng mga tao dito sa likod. Then we call this building the RNA loop kasi kung titingnan niyo yung structure na para siyang single-stranded na may loop, no, may hairpin loop. So the nickname of our MBB building is called the RNA Loop. And ang Philippine Genome Center, nasa likod nito. So, we started our facility dito sa second floor nitong building na ito, and it was still new. At maganda nga itong mga rails na, di ba, yung metal niya, parang molecular structure, nagkadikit-dikit, ano? So, we're very thankful to our architect conscious. So, ito yung mga tao sa PGC. We're a very small community ng mga researchers. In the beginning, nung nagsimula yung MBB, parang maximum 35 students a year lang. Ngayon, we can accept 50 students a year sa UPCAT. No? So ito yung MBB. So that was our first home. And gusto ko lang ipashare din sa ating audience, ano bang makikita sa loob ng PGC building. So I mentioned to you, mayroon tayong administrative wing on the left side where I am now, at saka yung ating laboratory wing noong sa right side. So we also learned to design a building that way from our experience and observations doon sa Academia Sinica sa Taipei, that they suggested to us na syempre mas iba yung wiring at piping ng isang lab. So mas mabuti na yung laboratory wing, ibigay mo sa isang, you assign it in a particular side of a building para mas tipid sa construction at mas efficient. So this is the right side of our building. This is our laboratory. So makikita ninyo yung mga clean bench, ito yung mga equipment, at saka ito mga routine equipment at ang mga mahal na equipment ng Philippine Genome Center na so this is all HEPA filtered, and grabe pala yung building sa laboratory, grabe yung air system, ang air handling unit, it's really very different from the usual university laboratories we have, no? So meron tayong ganyan, at ito doktora, ito yung mga examples ng mga investments ng ating government sa mga equipment na kaya nating gumawa nitong COVID sequencing ito. Marami nang tatanong, Dr. Ana, bakit daw 750 lang sinisequence namin? Di ba? Every week, bakit 750 lang? Actually, ang 750 na yan, malaki na yan. Kasi dati, isa-isa lang naman yung sinisequence natin. Ito nga, sabay-sabay 750 at a time. Or pwede naman din, of course, 3,000. So this is the Nova 66,000. This is a very powerful equipment and also very expensive, at least $1 million plus etc. So it's more than $1 million. At syempre, ang annual maintenance contract lang nito masyadong mahal din, ano? So we have that in the Philippines, and this is the one we use for COVID sequencing. This one, NovaSeq. This PacBio is actually also state-of-the-art equipment. Ito naman pang single molecule. Kung baga, tuloy-tuloy pwede mo siyang i-sequence yung genome. So, if you're going to sequence the human genome, ito dapat gamitin mo kasi yung ibang equipment, hindi niya kayanin yung laki ng human genome at saka yung coverage. So, gusto ko ipaalam, Dr. Ana, sa ating audience, so when we sequence the genome, for example, of SARS, or the human genome, or kung bacterial genome man, hindi yan siya sinisequence sa isang kopya lang. Normally, for example, for the SARS-CoV-2, we sequence that 1,000 times to 2,000 times kasi maigsi yung ating pag-sequence tapos ina-assemble yan sila ng computer. Yun yung gamit nitong mga equipment ng Illumina. Samantalang ito namang PacBio, which is a single molecule sequencer, kaya niyang i-sequence, for example, 10,000 to 20,000 bases sabay-sabay sa tuloy-tuloy, no? Depending on how you prepare the DNA. For example, hindi ba siya shared, yung putol-putol, basta intact siya, kaya niya sequence long. So, usually ito sila, when we assemble a genome ng mga organisms na complex, we do them together. Pero for the SARS-CoV-2 effort natin, ito lang yung NOVA 66000 that we use, no? So, and we use a COVID-sick kit. So, yan. Tapos, meron din naman tayong NextSeq 500. Ito naman ay ginagamit din natin kung mga 350 or 150 viruses lang gusto nating i-sequence sabay-sabay. Ginagamit din natin ito, and we have two equipment like this dito sa Philippine Genome Center. So actually, maraming tayong equipment, iba-iba, from the most powerful equipment to the medium, and then of course, so the simple one. So this one is MySeq FGX. We also use this, for example, kung mga 25, 50, or 100 lang ang aming i-sequence ng mga viruses at may kit kami doon. So tinawag siyang FGX kasi ginagamit din siya sa forensics DNA analysis. So yan yung mga equipment. So nakita niyo dito yung mga output, no? Output, ganyan. Tapos ito naman, Doktora, explain ko na ba ito o hindi muna?
Ma'am, ako muna magtatanong para...
Sige. Mamaya na lang. So, I will stop sharing muna. Okay. Yun lang yung story ng PGC.
Yes. Ma'am, congratulations. It's a beautiful story na talaga pong na-touch po ako that 4 women put up an advanced facility po. Of course, with a lot of help from the government and the UP po. So, this Philippine Genome Center is located in UP Diliman, ma'am.
Oo, we are near the National Science Complex. Yeah.
In the National Science Complex. Ma'am, question, saan galing yung funding nyo? From UP? Directly from the government?
Oh, yes. Yung support ng Philippine Genome Center, ang annual budget namin dito for our operations is supported by the University of the Philippines system. So yung Philippine Genome Center, yung Philippine Genome Center is directly under the Office of the Vice President for Academic Affairs and the Office of the President. Hindi kami under sa UP Diliman, although we are located in UP Diliman. Kumbaga, yung iba-ibang CU, kasama pa dito sa Philippine Genome Center. So we have that. And at the same time, Dr. Ana, since we also have services kasi may mga sequencing services kami, so we also have income from there. And also we have grants. We submit proposals to the DOST. And the DOST has been our champion. Very generous sila magbigay sa amin ng grant. And also all over the Philippines, yung mga researchers natin, kung may grant sila, di meron naman kaming customers, kumbaga. No? So, yun, that was very important.
This is really beautiful. Tapos yung sinabi niyo po, nung nagsara-sarado na po yung mga sarili-sariling scientist na lang, handa po ang Pilipinas for that eventuality. That is something that's really fabulous to learn, ma'am. And, sometimes we have a bad opinion po of the Philippines, pero kaya pala natin, we can take care of
At saka, Doktora, the fact na binigyan ng P.3, yung lineage na yun, naalala mo, it means that our young bioinformaticians can stand shoulder to shoulder with the best bioinformaticians in the world. Sila lahat gumawa na, yung mga bata na halos hindi na natutulog. So, Dr. Ana, di ba, meron tayong mga molecular biologists. Di ba, meron tayong molecular biologists. So, yung journey niyan, meron tayong mga registered medtechs. Nagtatrabaho yan sila sa biosafety level 2 plus facility para sa RT-PCR. Tapos, meron naman tayong isang team na naman, ito naman mga gumagawa ng sequencing itself. After na makuha yung sequencing na yan, matrabaho ito. It's really matrabaho.
Tanongin natin, why do we need, balik tayo sa basic, ma'am, kasi talagang, why do we need to sequence? Yun po. And then, paano po, let's say, if yung process po, let's say may naging positive.
Mm-mm.
Ilan po dun sa mga positive ang sinisequence po natin? What is gene sequencing? Bakit po siya mahalaga ngayon sa panahon na to? And ilan po dun sa mga, where do you get the positive? Do the lab send it to you? Hindi ko, what is the process po, ma'am?
Oo,
Sige po.
actually, Doktora Ana, kanina meron ako sa PowerPoint ko yung slide ko no na I can share the screen
Sige, ma'am.
again. Oo, that
Ipakita niyo
would,
po.
oo, because
We
that
are
one
so
would be
curious. What is gene sequencing? How do we move? Kasi after the positive, parang hanggang doon na lang po ang alam ng tao eh. But actually, may
correct.
ito pa po yun.
Process
What
ito, oo.
Opo.
So, Doktora, we were tasked, no, so the Philippine government formed a task force called the IATF Technical Working Group on COVID Variants. At bakit ginawa ito? Kasi naalala nila towards the end of 2020, lumabas na yung B.1.1.7. Ang hirap nga kasi, syempre, hindi maganda na sasabihin natin yung place of origin, no? So, this was in Kent, UK. So, palagi natin sinasabi yung B.1.1.7. Dinidiscourage talaga tayo ng WHO and other agencies na huwag natin lagyan ng place attribution. Ang problema lang, si WHO hindi pa rin nakagawa ng nomenclature system na madali maintindihan, maalala. So hanggang ngayon wala pa, kaya ang mga tao at ang media palagi nagsasabi ng UK variant kasi
Mas
ang
madali
hirap
kasi
din naman, oo, ang hirap din naman mag-memorize ng B.1.1.7, di ba? Ang hirap. So anyway, we have this task force, no? So sila yung nagde-decide, na kasama din ako doon sa task force na ito, nagde-decide ng methodology, and this is always done in coordination with the DOH Epidemiology Bureau. So sa Epidemiology Bureau, ang ating chief epidemiologist dyan, si Dr. Althea de Guzman. So we work closely with the Epidemiology Bureau. So ang ating DOH, nagpadala na ng notice sa ating mga different labs around the Philippines, yung mga testing labs natin. And I think we have more than 200 by now, na magpadala sila sa Philippine Genome Center ng samples na nakapasok doon sa criteria. Ano yung criteria natin? Number one, we need to have, may na-assign na mga percentage kung baga ilan ang samples na ipapadala ng bawat rehiyon. Mayroon tayong ganong numero. Pagkatapos, based yan siguro sa population, that was determined by our statisticians here and the task force. Pagkatapos, nilagay din doon sa memo na yun kung paano sila magpadala at ano yung qualifications. Number one, syempre, kailangan makipag-coordinate sa mga regional epidemiology and surveillance units. Number two, yung ating mga swabs na nag-positive, dapat yung city values nila below 30. Bakit below 30? Kasi sa aming validation dito sa Philippine Genome Center, and this was also similarly observed by other labs around the world, pag mataas pa sa city value na 30 yung isang RT-PCR, parang kulang yung ating target viruses doon at hindi siya ma-assemble ng mabuti ang genome. So, since ang ating purpose dito sa genomic biosurveillance is not putol-putol na sequence, gusto natin buong sequence ma-assemble natin, it's important na we have to have enough genetic material. So, ang ating requirement, the city value should be below 30. Tapos, pag-coordinate na yan sila sa mga resus at saka mga local government units. So, every time na kontakin kami dito, we always tell them to please coordinate with your regional epidemiology surveillance units that will also coordinate with our DOH Epidemiology Bureau. So, yun, ipadala nila sa atin. So, it depends on the condition. Pwedeng naka-dry ice or pwedeng kung fresh na fresh, pwedeng dalhin dito sa Philippine Genome Center agad. Tapos, meron din tayong mga hinahanap, for example, returning overseas Filipino workers, mga ROFs natin sa airport. We also sequence those. Tapos, yung mga bawat rehiyon, di ba, may mga na-allotted slots sa kanila. Kaso nga lang kung minsan meron tayong mga spikes of cases, may mga reports of surges, so doon parang binibigyan natin ng puwang na mas maraming ipapadala sila sa atin. At saka syempre meron din namang challenges yung much distant regions ng ating bansa na kaunti pa lang napapadala sa atin. So we're trying to polish that also with the DOH, the Epidemiology Bureau, kasi kailangan nating magawa yung parang national map that's national map or a national idea of the spread of the different variants. Tapos, Doktora Ana, pag sinabi natin variants, just because it's called a variant, it doesn't mean it's all negative, di ba? Tinatawag din siya ng variant kasi nag-accumulate na siya ng mga mutations that makes it different from the original source. So for example, ano ba yung ating original, yung the one that was first isolated in Wuhan, China, yun yung ating kumbaga reference. Tapos, yung mga variants na ito, syempre, because it's the normal evolution of a virus to mutate, so nagkakaroon na siya ng mga iba-ibang groups of mutations until such time that it might behave differently. So binibigyan siya ng lineage assignment, which I'm going to explain to you maya-maya. So ito na, received samples, tapos kinu-QC natin yan kung maganda yung pagkabalot, kumpleto yung information, tapos tama ba yung city values niya, tapos gumagawa na tayo ng whole genome sequencing. So, for example, what is the challenge in whole genome sequencing? Siyempre, the expertise is much more rigorous on process compared to RT-PCR. No? Kasi ang RT-PCR, extract the RNA, tapos mag-PCR ka, tapos makita mo yung curves, that's it. Ito, nagsimula ka dyan sa mga RT-PCR or sa reverse transcriptase, reverse transcription polymerase chain reaction. Pagkatapos noon, mag-PCR ka ng napakarami kasi kung ginagawa natin dito is really to target the virus. Why do we do that? Di ba pag mag-swab tayo, sa swab na yun, kasama doon human DNA, human RNA, bacteria, at kung ano-ano pa. So it's really a matrix of so many materials and so many genetic materials or sabihin natin contaminants. So if you want to sequence the virus only, kailangan natin i-enrich ang samples natin. So kung baga padamihin natin yung mga virus-specific sequences para sila yung ma-sequence natin at hindi yung mga bacteria doon, no? So yun, gumagawa tayo ng maraming PCR amplification. So yung suggestion nga sa amin, dagdagan pa namin yung PCR machines namin dito. We have how many? 4 to 6. Ang target namin 12 to 16 PCR machines. Hindi yung real time ha, just for the sequencing alone for COVID. So tinutulungan naman tayo ng DOH so that we can have additional PCR machines. Pagkatapos noon, Dr. Ana, pag makuha na ang sequence na yan, after so many rounds of PCR, tapos isa lang na doon sa machine na sabi ko yung Nova 66000 na machine, probably that will take about 12 hours. Pero yung PCR matagal kasi 750 ba naman yung ating samples and it has to be done very, very carefully. And then yung ipasa na yung sequences natin doon sa ating team sa Bioinformatics Core Facility. So yung ating mga bioinformaticians, they come from different disciplines, lahat sila bata. May mga molecular biologists, may mga agricultural biotechnologists, may mga computer scientists, at may mga physicists sa team na ito. No? So, magbuti, Doktora Ana, by the time na lumabas itong COVID, naka-assemble na tayo ng ganung team of people of various expertise na mahilig sila mag-computer work. Tsaka may biology background din sila. So before, I thought it will take more than many hours. Pero automate na nila yung a lot of the processes. So immediately within 5 hours, I think, the moment we receive it, we can immediately give it to the DOH Epidemiology Bureau na sabi namin, ito na yung lineages nila. So that is just the first part. Pero tapos syempre si DOH naman, magpa-press conference siya and they will of course, bago mag-press conference at ipaanam sa mga tao, nag-contact tracing na sila para naman malaman natin kung sino itong mga individuals na may mga variants of concern. Yung variants of concern, ang una talagang kina-contact tracing. Pagkatapos niyan, dito sa PGC, dito sa kabilang wing ng mga focus computers sa Bioinformatics Core Facility, gumagawa pa tayo ng marami pang analysis, detection of mutations, rare and variants. So these are all computational work, some of which are nagsulat sila ng mga script para ma-analyze ito, no? Yung lineages, pwede na yung ma-automate. Tapos merong pangguling, which I'm going to describe mamaya. So may phylogenetic analysis, phylodynamic analysis through time, haplotypes, tapos mga variants of concern, tsaka kung paano namin nakita yung P.3, no? Kasi tinitingnan namin yung mga mutations sa SARS-CoV-2, particularly those in the spike region. So yan na. So ito yung process. Dr. Ana, gusto mong i-describe ko ito in simple terms? Yung
Opo.
sinasabi ko kanina sa inyo, no? So may sample, tapos marami tayong primers. Essentially, yung primers, pooled primers yan. Yung ating SARS-CoV-2 RNA, kasi it's an RNA virus, is about 30,000 bases. So from an RNA, kailangan siya i-reverse transcribe into a DNA. So using a reverse transcriptase. So yun, meron tayong reverse transcription polymerase chain reaction. First time yan. Tapos pag DNA na siya, multiple na yan, i-enrich na natin yung samples natin using primers na overlapping so that we can assemble the 30,000 bases ng virus DNA na, no? Virus DNA. And of course, di ba, naalala mo, we are doing it 750 at a time or pwede na bang 350, 750. There are actually multiples of 384. Tapos, the reason that we say 750 kasi yung iba doon mga controls, no? Kasi we have to have internal controls for RNA kung tama ba yung ating processes na ginagawa. So there's always an internal control and positive and negative controls. So ito na yon. Tapos lahat ng yan, yung 750 samples, for example, lahat sila naka-barcode. Ibig sabihin, bawat sample na yan, meron siyang sariling overhangs na naka-barcode sa kanya para ito si patient A, si patient B, si patient C, which we demultiplex later on sa computers na. No? So bawat sequences nitong sample A na to, na maraming gina-assemble na to, meron siyang barcode. So lahat sa kanya, like XYZ, XYZ, XYZ. So kung baga meron tayong marker para sa kanya. Yan. Tapos ipupull na yan at saka iraran dito sa ating machines is either Nova 66000, a NextSeq 500, or an Illumina MySeq. It depends, for example, on the number of samples we have. Kasi ito pinaka-powerful ito. Ito naman kaya 350, tapos yung MySeq kaya 25, 50, or probably 100. So, tapos, paano natin malalaman ngayon yung mga sequences na yon? Paano natin agad na-assign ng lineages at naibibigay agad sa Epidemiology Bureau, no? na na-announce sa ating mga media people at mga tagapakinig at nire-relay sa ating mga sending RESUs or Regional Epidemiology Surveillance Unit. So there's such a thing as a website called GSAID. So this is the database. GSAID was actually, of course, an offshoot of the efforts of many scientists to share data. So ano ibig sabihin ng GSAID?
Ma'am, yan yung original SARS-CoV.
Oo, so GSAID, it is the, nagawa ito yung original. Tapos ang mga influenza, yan. May
nandyan po yung original. Tapos nagsisequence po kayo ng mga DNA, yung galing po sa Pilipinas. Opo.
Tapos pinapasok namin yan lahat. Oo.
Or kung
Doktora Ana, Doktora Ana, itong GSAID, which is the Global Initiative for the Sharing of All Influenza Data, it's really an org. It's an org started by scientists and with the help in the beginning of the German government. And also, ang dami, unprecedented. I think SARS-CoV-2 is the most sequenced organism in the world. So, nagsimula sa kaunti lang hanggang ngayon mga 700,000 na yung na-deposit sa database na ito at siya ang nag-a-assign ng lineages, na mayroon siyang app, yung pangolin.
So, bawat country po right now is sequencing also. And then, sinasubmit po nila doon para malaman.
Sa GSAID, ang GSAID pala meron siyang nomenclature system. At saka meron din tayong EpiCoV database. Ito pala yung pangolin. Na-notice niyo kasi yung United Kingdom, they have a network. They call it the COG, which is the SARS-CoV-2, the Coronavirus Genomics Consortium sa England. Practically, Dr. Ana, mga 50% ng contributions dito sa GSAID database came from the UK. Kaya pag sinabi nga natin, Dr. Ana, na UK variant, actually maraming UK variants kasi si UK ang ano niya, masipag siyang magsubmit ng sequences dito, no? So we have this database, yung GSAID na to. So tayo sa Pilipinas, we started submitting in April dahil nga tayo nag-sequence tayo doon sa UP kit, di ba? At saka MTech kit, we submitted the sequences there. So ito na yon. So yung may pangolin is is this siya yung nag-a-assign ng mga numero ng lineages ito. serum bottom. So, lineages A.1, B.3, B.1.1, ito yun. So, ang pangolin is called Phylogenetic Assignment of Named Global Outbreak Lineages. So, sa pangolin website, may app ito, yung EpiCoV database, doon tayo. So, naka-link na tayo dyan. So, every time we have a sequence, na-download na natin yan, we can immediately assign them to the database. And it keeps on getting updated. Yung P.3 nga, hindi pa nila na-update, pero sa pangolin sa kabila, na nakita na nila yung P.3, pero yung app nila na automatic assign ka sa P.3, hindi pa nila na-update. Siguro next update nila. Na? Tapos ito naman, may next strain, may disease. So may maraming mga database. Kaya nga, alam mo, sa kadami-daming sequences, I think they have to come up with a much better nomenclature system ngayon. Kasi for the experts, okay lang magsabi ng B.1.1.7, but it is so difficult to understand for others, di ba? No? Kasi ang maganda naman dito, malalaman natin na for example si B.1.1 ang galing kay B.1. So in terms of the genetic sequence, malaman natin kung paano nanganak-nganak itong mga viruses na ito. So ito yung una nating work, Doktora Ana. Yung si Francis, ito yung ating chief bioinformaticians dyan. So palagi nga siya yung first author sa ating mga manuscripts. Ito yung ating unang effort na ma-detect yung mga sequences natin dito. So yung mga sequences na sequence namin dahil sa mga volunteers sa PGH in March, nakita namin na ang dami sa kanila nag-cluster sa Diamond Princess na mga submitted sequences din. So in the beginning, in January, we had some visitors from Wuhan, China. Ito yung mga nasa RITM, ano? So we have those. The second is, of course, yung Diamond Princess na ito. At saka yung June, ito yung sinabi natin may D614G mutation. Ito na yung D614G variant in June that came to us probably, I think it is safe to say na yung circulating viruses sa buong mundo, palaging mayroon ng ganito, yung D614G as a base mutation. So, we noticed that, so this is called a phylogenetic tree at makikita mo yung pagkabuo-buo nito. Actually, Dr. Ana, hindi ito mabilis gawin. This is a lot of computer work, no? So it takes days to come up with this one, lalo na kung libo-libong sequences ginagawa mo. Yan. So yan. So gusto ko lang ipakita sa inyo, for example, when we detected the one in Cordillera Administrative Region, makikita natin na UK variant. And dahil nga doon sa sequences niya, at when we align them with the sequences of other countries, we realize na meron siyang tail mutation na UK variant. So actually, itong sequences ito very, very relevant. Kasi you remember, we had 117 na nanggaling United Arab Emirates, nanggaling Lebanon, and we realized na yung kanyang pagka 117 UK variant, iba siya kaysa sa nakuha doon sa Bontoc cluster, which probably came from the UK. So, makikita mo ngayon na, mayroon pala kumbaga within the 117 variants, merong pang Middle East, merong pang ganito, pang ganyan. So, makikita mo doon sa sequence information. Para siya kumbaga may footprint or may fingerprint siya, no?
So,
Based on the
may
sequence.
variant po at may sub-variants nung variant na yon, depende kung saan nanggaling.
Oo, so for example, yung root nito, let's say nanggaling Wuhan, China, nanganak, at saka lumabas na yung UK variant, and within the UK variant, nag-mutate siya rin.
May mga anak. Opo,
may mga anak.
May sub-variant, yung variant.
May mga sub-variants na yan. So makikita mo, for example, na yung nanggaling sa samples ng United Arab Emirates or ng Dubai at Lebanon, magkalapit sila as opposed to sequences na nakuha natin doon sa Bontoc na malapit siya sa UK na mga samples. So makikita natin yan, no? So ganun.
So, opo.
So that is,
Opo, I'm just so interested po talaga. Balikan lang po natin para maintindihan po ng mga tao.
Okay.
The Philippine Genome Center, sila yung nagsisequence. The meaning of sequencing is alamin natin kung ano yung mga DNA and RNA ng bawat COVID.
Pagkadugtong-dugtong sa ating genetic material.
So, yung pagkadugtong po nila. Now, if there is a difference sa pagkadugtong, and if it's a significant difference, then that is what we call a variant.
Oo, kung baga, nag-accumulate na sila kasi yung mutation, Dr. Ana, pwedeng isa, pwedeng dalawa. Pero just because may mutation siya ng isa, hindi siya agad variant, hindi pa matawag agad ng variant.
Kailangan significant. Opo.
Correct. Tapos na-assign siya sa variant.
And then, kino-compare nyo po ito to a global database of variants in
Correct.
the original
In
COVID.
the
And then
world, oo.
sinasabit po abroad. Opo.
Oo.
And
So,
then, ano po, how many can you sequence a day, ma'am? And how
ah,
long does it take?
so it's really dependent on the machine you use, 'di ba? I remember Dr.
Ay,
Ana,
sabi
I
nga niyo
told
po,
you.
there are a number of machines. Opo. So,
Oo.
matagal po
Pag
yung
ito,
proseso.
oo,
Opo.
itong sa kit na ginagamit natin, which is also used by many people around the world, kung may machine silang ganito, kasi yung kit natin ginagamit, hindi mo pwedeng gamitin dito. Dapat sa ganitong machine lang. Kung ganitong machine, ibang kit na naman gagamitin mo.
Opo.
So, Dr. Ana, itong NovaSeq na ito, pwede kang mag-sequence dito 750 or actually it's 384 times 2 times 2 times 2 times 2. So, up to 3,052 at a time. The reason na hindi natin ginagamit, doktora, yung 3,052 at a time, sobrang tagal ang handling time ng samples sa laboratory. So, can you imagine, i-inspectionin mo 3,000 tubes, tapos i-extract mo 3,000 tubes, tapos targeted resequencing mo yung 3,000 na yun, paulit-ulit sa ilang PCR machines. Hindi maganda, hindi sulit yung kanyang turnaround time. So, tatagal siya. So, yung pinakamagandang balance is actually 750. We determined that to be 750. So, mga tatlong araw siya. Kaso nga lang kung
Mga
minsan siya,
3 days po.
3 days. So technically, dapat 30 hours to 48 hours. Dapat hindi siya mag-abot ng 72 hours. Sa atin kasi dito, syempre, nanggaling sila sa iba-ibang panig ng Pilipinas, kinukolekt pa natin sila, ini-inspect at tinitingnan kung tamang CT, and then from all the submissions, we prioritize which to choose. So for example, Dr. Ana, merong isang cluster na 30 sila. Hindi naman pwedeng 30 lahat isi-sequence mo, di ba? Kasi we also have to
Opo.
allot sequences. So we can get representative samples lang. So yung nagde-decide po
Opo.
niyan, yung
Bali,
ating
opo.
TWG.
Yes, ma'am. Sino nagsasubmit sa inyo, ma'am? The private labs, the
All
DOH?
labs, all labs.
All labs nagsasubmit.
Opo,
Opo.
opo. Ang
So,
nahirapan lang
how do
tayo,
you know which to
Dr.
prioritize, ma'am? Like yung mga surge siguro, no? And
Oo,
nabanggit niyo
they have to
Opo.
coordinate talaga, Dr. Ana. They have to coordinate with the RESU, with the Regional Epidemiology Surveillance Unit, at saka sa ating Epidemiology Bureau dito sa
And they will
DOH.
recommend certain groups na yun ang dapat
Correct, correct. Oo. So kung minsan,
Halimbawa,
doktora,
biglang nagkaroon ng surge, baka doon ho kayo titingin muna. Kasi
oo,
parang,
idagdagan namin yung samples. Isama namin sila, no?
opo,
In fact,
opo.
you remember last week ba? Last Monday ba yun? Last week Monday, we actually did a special run for National Capital Region only, no?
Para malaman
Kasi,
niyo po.
para mabili,
Opo.
oo. Kasi, Doktora Ana, pag let's say ang aming isi-sequence 50 lang o 25 lang, overnight malabas na namin agad yung sequence, ang result.
Opo, so it
Very
depends
fast.
kung ilan po ang iraran. Opo.
Correct. Oo. So tumatagal lang tayo kung minsan kahit 3 days yung actual hands-on, tumatagal siya ng 1 week kasi kinukumpuni pa natin yung mga submissions, dinidecide natin kung ano ang isama sa run na ito,
Opo.
gagamitin ba natin yung mga biobank samples o yung mga active cases lang. So that determination is actually based on the IATF, TWG together with
Opo. At saka based on the situation on the ground.
correct.
Tapos dito, yun ang uunahin nyo.
Tapos kung minsan doktora sasabihin ng Epidemiology Bureau na ang National Capital Region, we need to have let's say Pasay, remember? Pasay.
Opo, pag may
Oh,
emergency.
oo, grabe, 24 hours yan ginawa kahit, ilan lang ba yun? 50 samples lang yata yun eh. Ginawa namin yun overnight. So yung mga 25 lang, kaya namin yun gawan overnight kasi mabilis lang. Mahirap yung 750 kasi ang daming hands-on time.
Opo, it's
So,
time. Opo.
yeah. So, ginagawa namin ng doktora. So, usually, kung mga 50 lang, ito ginagamit naming machine, no? At saka, iba kasi yung kit dito. So, we actually buy several kits din, just in case. At saka pala, Doktora Ana, hindi ko na i-share sa mga tagapakinig natin, dahil nga sa necessity ng sequence, kasi di ba nag-RT-PCR na tayo? The RT-PCR tells us, for example, yes, no, di ba?
Yes,
Pero,
no lang.
yes, no lang siya.
And
Yes,
after
no lang
that,
siya.
ano yun.
Correct. Of course, kung may kit, for example, na yung TACPath na yung kit, pwedeng kung may walang S na ma-amplify, pwedeng most likely UK variant. Ano? Pero yung ating RT-PCR is just yes, no. If we want to know the variants, we really have to sequence.
We have to sequence.
Oo. At saka hindi rin pwedeng i-sequence mo lang. That's may iba tayong sequencing machine na yung capillary sequencing. Yung isang region lang, ang daming mutations bawat variant, di ba? So, kailangan ka talaga mag-whole genome sequencing. Now, nung nagsabi na yung mga WHO na kailangan tayong mag-whole genome sequencing, at nakita natin kasi syempre natatakot lahat dahil sa mga variants of concern, naku, nagsibilihan sila ng kit, nagkaubusan ng kit. At hindi lang ang kit. Yung iba-iba pang mga materials kasama sa kit kasi may sequencing kit pero meron pa tayong mga pinag-QC na mga materials. Naku, nagkakaubusan imbis na i-deliver na 1 month delay, 2 months delay, ganon, grabe.
Nagkaubusan po globally.
Oo, nagkaubusan globally at saka yung not only for the
the
sequencing kit but for the other reagents. Correct.
because everyone is sequencing all over the world ngayon.
Correct. Everyone is sequencing all over the world.
That we have our own lab. Otherwise, aasa lang tayo sa ibang tao.
Tsaka doktora, ang hirap-hirap kasi their own countries are under pressure to come up. Can you imagine this?
So it's very important to have your own lab.
You have to have your own lab. Oo. And we train more people. We train more people na matuto nino. So actually, doktora, may mga laboratories din sa Pilipinas na meron silang itong machine na ito, na ginagamit nila sa TB or sa dengue, for example, kasi yung mga viruses, maliliit lang yung genome size nila. So kayang-kaya sa ditong machine na ito, no? So may mga labs na rin sa atin nag-approach at we are all on memorandum of agreement na itulungan namin i-train ang staff nila. Hindi ko lang maisabi kung anong mga hospitals ito na may ganito kasi gusto din nila mag-sequence. So sabi namin, it is part of our role as the Philippine Genome Center. Of course, we are in UP but our reach and our breadth is beyond UP to capacitate others. So we are also in the stage where we are also going to train other hospitals na may ganitong machine if they want to send to people to the Philippine Genome Center, of course, with all these biosafety protocols. No? So merong mga labs, Doktora Ana, sa Pilipinas na may ganitong machine kasi perfect ito sa yung mga pathogens, yung mga maliliit. So kaya niya mga 25 to 50 at a time, kaya niya. So may mga hospitals na may ganito. So sabi namin, we can help train you at the same time because marami na kami experience on how to fine-tune the protocols. So meron din na may mga labs na we are in consultation now na pwede na rin mag-offer nito at least for their own samples in the near future. No?
Ayan.
Yeah.
May tanong po dito, please tell us about the Philippine variant at paano nyo po na-discover?
Sasabihin ko yan sa susunod na slide. Doktora, pwede na ako mag-segue doon?
Yes,
okay, so ito na yung ating ganito. So you remember we assign lineages, pero yung Philippine variant, hindi siya madali makita kung wala kang suspicion. Kung wala kang suspicion kasi pag assign ng lineages, na-assign siya sa B.1.1.28 which is the usual, hindi yung variant of concern, the usual Brazil variant. Pero sinasabi ko sa team natin, sa bioinformatics team na pinadala sa atin ito ng Central Visayas, there must be something there. Kasi dati, yung B.1.1.28 na yan, hindi naman yan common sa Pilipinas. Narinig naman ninyo yung DOH natin, palagi nagsasabi na yung common sa atin, yung mga Hong Kong variant at saka UAE variant. Common talaga yon in the early days ng ating sequencing. Tapos all of a sudden, we had this number of samples coming from Region 7 and they said that they have a spike of cases. Tapos ang lumabas ng sinequence na namin, B.1.1.28. So pag ganun lang, parang known variant siya. But what I did was, ito naman kasing ating mga bioinformaticians, we are always in communication all throughout the day. Kung i-text ko sila alas 6, may sumasagot. Kung alas 9 ng gabi, alas 2 ng umaga, may sumasagot din. But anyway, doktora, ginawa namin yan. We did mutation analysis of the entire sample set na nakuha namin doon. And then we realized na, my mutations sila. And then we inisa-isa sila lahat until we came up with this mutation profile. No? So I'm going to share this to your audience now. So this is what we did. So essentially...
Sorry, basahin ko lang po yung tanong. 1 second. May tanong po dito. Paano po nahanap ang bagong Philippines variant? Wag Philippines, okay? P3, guys.
P.3. Ito yung nahanap namin. So yung nahanap namin ito na pinapakita ko na sa inyo.
Sige po.
So this is the answer to that question. So
Sige po.
there was a spike of cases in Central Visayas, and they sent to us these the samples that they have. And when you do a pangolin, kasi wala pa P.3 noon eh. When you do the pangolin database, they were assigned to the B.1.1.1.7. Doon sila, B.1.1.28, doon sila na-assign. Pero sabi ko, parang hindi siya, parang unusual, bakit marami doon sa Region 7 yung kanyang variant. And abi, in mutation analysis nyo, isa-isahin nyo yung bawat sample, bawat nucleic acid, kung ano yung mayroon siya. Doon namin nakita na may mutations pala sila beyond the B.1.1.28. Kaya kailangan, hindi lang pwede na lineage assignment lang, kasi kung lineage lang, hindi mo ma-detect agad na meron pala siyang mga mutations included there. Kaya nagkaroon tayo ng P.3 dahil sa ating nadiskubrihan na yung nakikita nating B.1.1.28, meron pa palang additional mutations other than that background. So, ayun yon. Ito yan. Nung nakita sa akin, ako, syempre, doktora, we have been watching kasi that is our job in the genomic biosurveillance ng ating IATF to detect all these and to tell our Department of Health na we are looking at these potential variants. Oo. So nakita natin ito noong February when we did our sequencing run on February 4. Nakita natin na beyond the assignment of the B.1.128, nakita natin na meron siyang 2 mutations. Original, yung 484 at 501 lang muna yung sinabi sa akin ng team. In less than 24 hours, sinabi nila, ma'am, hindi lang dalawa, marami pa, ito pa. So what are these? I'm going to explain very, very slowly and very carefully. Let us assume that this one is sequence nucleic acid number, letter ba kung baga sa RNA o sa DNA, number 1. Ito, 30,000. Ito yung complete genome ng virus, no? Assuming. Within the genome of the virus, there are several genes, ORF1AB, ginagamit natin yan palagi sa ating sequencing RT-PCR. The S, the pula na ito, is the spike region, and everybody is concerned about the spike kasi many of our viruses, many of our vaccines were designed based on the original sequence of the spike region of the SARS-CoV-2 virus that was published or shared to the world by China last January 10, 2020. So, ang dami nating vaccines were based on the original spike region. At saka bakit spike region? Kasi yung spike region ng virus, yun yung nagko-contact sa kanyang receptor sa human cell na ACE2. So, this has a DNA or binding site, sa receptor binding site. So, importante siya. Tapos, iba-ibang mga genes, mga N, mga M, E is envelope protein, M is of course, M ba to? Membrane protein, N is the nucleocapsid at different kinds. So, for our purposes, nag-focus tayo sa letter S, itong spike region na ito. Tapos, ni-map na itong mga bata, itong mga bioinformaticians natin, saan ba mga mutations ng P.1, ng B.1.17 that was first discovered in the UK, and the B.1.351 that was first reported by South Africa. So sa ilalim nakita ninyo may green. Ito yung mga locations ng mutations nitong mga variants of concern. So ginamap natin yan. Tapos vis-a-vis this location, tinatingnan natin kung yung mga mutations na nakikita natin sa mga Central Visayas samples, at hindi lang Central Visayas, entire Philippines, nagpupunta dito sa mga region na to. So, for example, the N501Y, o diba, pareho siya sa lahat. This is a shared mutation by all the variants of concern. The one from UK, South Africa, and the one from Brazil. And then the E484K is shared by the South African and the Brazil, quote-unquote, variant, with apologies to South Africa and Brazil, no? Yung 484K. So nakita natin agad, kaya sinasabi ng DOH na mayroong co-occurrence ng dalawang mutations. But actually, we realize that beyond these, there are other mutations, and this is one is quite significant. Meron siyang tatlong amino acid deletions in positions 141, 142, 143. Samantalang yung UK, meron siyang isang deletion. Ito UK. Ang UK may deletion in amino acid 144. Ang Philippine samples na nakita natin, ang mutations niya tatlo, hindi isa. Tatlong amino acids on the left. on the left, no? Of the one from UK. So, there must be some significance into this region, di ba? Tapos, may iba pa, yung ibang mga hallmark mutations. So, pinaalam natin yan sa Department of Health at pinaalam din natin yan doon sa ating mga teams ng DOH sa Cebu na may nakikita tayong ganito, and because of the potential clinical and functional implications of the mutations that we're looking at, it is very, very important that we do up to the third generation tracing, that's number one, and that we look very closely and follow them. So, maganda naman, Dr. Ana, sa lahat ng samples, 33 una, pero we now have 104. Sa 33 na ito, lahat sila puros mild or asymptomatic yung ating mga samples coming from Central Visayas. So, this has not been associated with a more severe symptom, but nakita lang natin siya na nakita natin siya sa isang spike of cases doon sa Central Visayas. So, yun, napaalam natin yung February pa doon sa Central Visayas na mayroon tayong co-occurrence of mutations until such time that we were allowed by the Department of Health to upload our sequences to the GSA database. And then we also submitted the manuscript. And when we uploaded the manuscript sa database, of course, we were suspicious, Dr. Ana, na it's going to be P.3. But we cannot say P.3 kasi hindi naman tayo ang nag-a-assign ng numero, di ba? We were already thinking na most likely it's a P.3, but we cannot assign it. So we just called it Philippine B.1.1.28. So I'm going to tell you, this is the list of those mutations. Ito, Doktora, bakit namin alam na I think it's going to be a different lineage? This one, ginawa nito mga computer scientists natin sa taas, yung mga bioinformaticians natin. How were we confident with a certain level, to a certain level na iba siya? Bakit alam namin na it's going to be
iba pa? Opo.
Paano namin nalaman na iba siya at paano namin nalaman na most likely maging P.3 siya? Actually, sa manuscript namin, in the beginning, nilagay namin P.3 pero ni-remove namin, in-erase namin yung manuscript kasi hinihintay namin na ang pangolin mismo ang mag-assign sa kanya ng P.3. So, when it was assigned, ikot na ito, the moment it was assigned sa database, we immediately knew na, finally na-assign na siya ng P.3. That was March 10. We submitted our sequences March 3. So we were waiting every day kung ma-assign siya ng P.3. We have been waiting since March 3 until March 10 na assign na siya. Okay, so paano namin, Doktora, nalaman na it's going to be a new name or a new variant? Okay.
Bakit sub-variant lang, ma'am?
Oo, bakit hindi siya sub-variant lang? Correct. Oo. How did we know that it's going to be one? Kasi that was our contention in the paper that we wrote. Okay. Itong ating mga bioinformatics, nag-download sila sa database and download nila from GSAID yung mga B.1.1.20. This is called a Brazil variant also, pero this is not a variant of concern, but it's also called Brazil variant, but not a variant of concern. So the B.1.28, ito yung mga yellow na yan. So yung iba dito, nanggaling Pilipinas, at ang iba, nanggaling sa buong mundo. Tapos meron tayong B117, B117, diba? Ito yung originally described or reported by the UK. Ito yung purple, ito yan. Tapos ito hindi yon sa Pilipinas, ha? Nakunuha natin yan sa database. Yung iba, of course, nasa Pilipinas. Yung P.1, this one, the orange, ito yung Brazil variant of concern. So tinawag siyang P.1 kasi actually, ang P.1 is a B.1.28.1. Kaso hindi pwede 4 ang numero. Kaya ginawa na lang siya P.1, which is actually an anak of the 28. So did you get it? Na yung P.1 is actually B.1.1.28.1. Yun yan. Yan. Kaso sobrang haba yung kanyang pangalan, may nickname na lang siya, binigyan na lang siya ng P.1. Okay. Ang Brazil, nagsabi din siya na may P.2. Yung P.2 is a variant under investigation, hindi pa siya variant of concern. Merong mga reports na probably in reinfection, etc., etc. Pero yung variant of concern lang dito, ito, 1, 2, ay hindi ko pa nalagay ang South African variant kasi at the time we were making this, wala pa naman tayong na-observe at na-detect na South African variant. So, Doktora, nakita mo yung P.2. Ito yun sila. Anak rin sila nitong B.1.1.28. Puro sila anak nito na P.1 and P.2. Yung ating samples na na-detect doon originally sa Central Visayas, na nauna nating na-detect from samples coming from Central Visayas, monophyletically, kung baga iba siyang branch. O tingnan mo, separate. It's not. Yan. So, it's a monophyletic tree. And in the same way that you have a P.1, you have a P.2, most likely this is a P.3. So we were suspicious that it will be named P.3, and so we waited for it to be officially assigned a P.3, and that was on March 10. So alam namin, Doktora, na ganyan siya.
Kasi iba siya. Opo, iba siya.
Oo, and we do this thing. So this one is not trivial to do kasi malaking computational work ito. This one, come up with this one. Mayroon naman ding next trend. So nakita mo rin, Doktora, this is a phylogenetic tree kasi through time, April, July, October, January 2021. And you notice sa atin sa dulo kasi syempre, nakita lang natin ito January, February sa ating samples. And by the way, Doktora, it was very interesting kasi when it was a share, it was assigned a P.3. Merong isa doon, 33 na samples coming from us were given a P.3. So nakita namin yung mga conversations ng experts na this deserves a P.3. Tapos, any objections? May mga ganon discussion sa pangolin. Nakita namin, Doktora, meron din doon nakasama na isang German submission. So 33 from the Philippines and one submission from Germany that was collected, I think, May 11. When we did, we don't have the data kasi hindi naman nag-press conference Germany as opposed to Japan na nag-press conference, may press release. When we looked at the sequences of the German, the Japanese, ang source root niya, Doktora, pareho sa samples natin ito. So most likely they are also rooted in our Central Visayas samples based on sequence information.
So ma'am, so basically, the Philippines discovered the P.3, hindi po Japan.
Sure. Hindi. Huling-huli na yung Japan. March 10 na yan. By that time, it was already given a P.3 name. Na-publish na ito online March 6. Ang Japan, March 10.
I would like to say, the Philippines discovered the P.3.
Syempre. At saka, Doktora, it's also a triumph of our effort, of our government's effort in genomic biosurveillance. So, nang sinusulat namin at sinasabit namin ito, parang sabi namin, dito sa Philippine Genome Center at sa team na rin natin sa IATF, na it was really like a turning point in the lives of so many of these people. And forever, basta P.3, magiging Tablizo et al. Na yan kasi siya yung sinasite palagi na sa P.3. This the first author.
Ma'am, para maintindihan po ng mga tao, tell me if I got the sequencing or the sequence of events correct. Kasi may nagsabi dito, yung simple lang. So parang dumami ho yung kaso sa Central Visayas. So, sinabmit po sa inyo yung sample.
Oo, as in, in-aeroplano yun, in-aeroplano.
Nakusap po sila, ma'am, please investigate Central Visayas dahil dumadami.
Yes. So, General Mel Feliciano.
Mga galing sa Central Visayas.
Correct.
So, pag po, nalaman nyo na hindi na siya yung
in the beginning, Doktora, hindi mo siya malaman. Na-assign lang siya ng B.1.1.28, yung parang mga yellow kanina. Pero,
yun po yung
yung una. Pero
study, nalaman nyo na maraming bagong characteristics.
Correct. In the beginning, Doktora, dalawa lang muna yung nakita namin. Yung 501 lang. Actually, in less than 24 hours, lumabas na, bumalik na sa akin yung ating mga bioinformatician na, ma'am, hindi lang dalawa, ma'am. Probably 13. Hallmark.
So there were 13 differences from the existing.
Correct. Ito yan sila, Doktora. These are all the differences.
So yung differences po ba? It's in the, yan ho yung 13 differences. So sobrang dami na ho ng differences na significant na po. That's when you learned na it was already a new, hindi na siya yung dating umiikot pero may bago na po.
At the same time, Doktora, importante yung yung phylogenetic tree. Kasi pwede naman siyang...
And then you found out from the tree that it was different from the previous.
It's a monophyletic clade, yes.
Tapos sinubmit yung abroad and it was accepted as a new variant.
Correct, correct. So they
So
always
yeah,
cite
so that
Tablizo
really
et al.
talaga kasi
So,
um,
ginawa natin yun ng buong Pilipinas, mga Pilipino lahat gumawa.
yeah, so
And
that's
that
really a
means,
scientific triumph.
that means, Doktora, ang ating bioinformatics analysis is very robust
Opo.
because our
So basically,
sequences were accepted, the mutations are correct, accepted sila, tapos
tama po
because
yung
of that,
proseso talaga. Yes,
tama ang
ma'am.
proseso, dapat ang pag-analyze,
And
no? Yeah.
internationally accepted. Opo.
Correct, correct. Ito, Doktora, kasama yan sa manuscript natin
Opo.
na
And the Philippines discovered this variant po in
from
Central Luzon.
ourselves. Yeah, oo. And
Ma'am,
doktora,
opo.
gusto ko lang isabi doktora na the more we sequence, the possibility of us looking for other variants is always there. They may not be variants under investigation kasi pwede namang wala sila yung 501 and 484, pero pwede tayong gumawa ng kumbaga local version ng kung ano-anong lineage na ito dahil nga nag-mutate na yung virus. Kaya importante na hindi dapat mag-increase ang transmission. The more transmission we have, the more possibility na mag-mutate talaga ang virus. And they will get local character, doktora, kasi pag siya yung nag-iikot-ikot sa ganyang lugar, malalaman mo tuloy na, oh, let's say ito, ganitong rehiyon, ganitong rehiyon. Well, we are actually observing that also. So, for example, doktora, meron tayong mga B.1.1.28. Kahit pareho silang lineage, makikita mo na sa ganitong rehiyon, may grupo sila. Sa ganitong rehiyon, may grupo sila. Pero hindi ko pa ma-share yan sa inyo ngayon. Pero that happens. We are looking at it.
To ask this, ano po yung B at saka ano yung P? Bakit may B at may P? Is there
Okay.
some significance or
Okay.
wala? Hindi ko rin
Oo.
alam.
O sige, ang una, in the beginning, yung pinaka-original virus that was described by China, letter A yun.
Okay.
Letter A. Tapos, nung nakita na natin, so may A.1, A.1.2, may mga ganon. Now, nung nakita na, you remember, nagkaroon ng D614G mutation?
Yes, ma'am. Yun yung pumunta sa
yun na nagpuros ayan sila B. Nagpunta na tayo sa B because of that one. Let me see. So, for example, ito na, yung original na yan na letter A, itong A, yan yung original na nanggaling sa first described by Chinese researchers centering on the sample centering on Wuhan, China. Pagkatapos nag-letter B na, kasi nga nag-mutate na siya. So itong mga B na ito, Doktora, B.1, pero na yan silang mga mutation sa B614G. Nag-mutate na sila. So puros na B. Tapos ano naman yung letter P, di ba? Tanong ni Doktora, ano naman yung letter P? So actually, yung P, Doktora, ganito. Nang lumabas itong si Orange, ito, anak siya ni B.1.1.28, anak siya. Ito, nag-branch siya. Monophyletic lead. So, kung baga, ang pangalan sana ni B.1 is B.1.1.28.1. Kaso sobrang haba ang 0.1 na naman, 4 na numero na. So they gave it a name P.1. Ito. So this is actually rooted in a B.1. Kaya yung Philippines, Doktora, yung una nating na-discover doon sa Central Visayas, in the beginning, nilagyan na lang natin ng Philippines-B1128 kasi hindi pa siya nabigyan ng P.3 na nomenclature. Wala pa.
So, yun po yung nomenclature po.
Oo. So, ngayon, Doktora, i-change na namin ito. Gagawin na namin itong P.3 sa manuscript.
Apo, P.3. Ma'am, question. Prior to the UK, sorry kasi hindi ko na maalala yung mga numbers UK, South Africa, at Brazil. Ano po yung dominant variant na umiikot like nung November?
Sa Pilipinas, oo. Ang nakita natin, Doktora, at palaging sinasabi ito ni, actually na-share na ito na statistics ng ating DOH Epidemiology Bureau, ang pinakamarami natin, Doktora, yung dalawa, yung HK, Hong Kong variant, at saka yung UAE variant. Pinakamarami sila. UAE, United Arab Emirates. I think, I cannot remember, baka ma-interchange ko. B.1.1.1.63, yeah, B.1.1.63 at saka B.1.263. Parang dalawa sila, Doktora. They are the most dominant even now in mass. If if we just if you just make a denominator since the beginning of time, yung Hong Kong variant at ang UK variant ang pinakamarami sa Pilipinas. Pero we have to study it very closely, doktora, kasi as variants of concern evolve, hindi natin alam kung sila magdami o hindi, no? So yung dominant variant is a function of time.
Opo,
It's
kasi
really a function
nagpapalit
of time.
po po. Depende
Correct,
kung ano
correct. Pero yung
nangingibabaw. Opo.
correct. So sa
But
ibang
nung
lugar ng
nakaka
Pilipinas,
po, it was the Hong Kong and UAE variant po.
yeah, even now, so for example,
Up
itong ating
now
variants of
po.
concern, mga in total, I think we have sequenced more than 4,000, no? Mga more than
Opo.
4,000 na sequence natin since January 4. Yung in total, parang yung ating mga variants of concern na ito parang mga less than 10% of the entire sample set. Pero yung mga yun, marami yan sila. Let's say 20%, 15%. I don't have the exact values, pero si Epidemiology Bureau has the statistics na ito ang pinakamarami sa Pilipinas, yung Hong Kong variant at ang UAE variant.
Yun talaga yung dominant po.
Sa atin, oo, let's say sa mga Cagayan Valley, let's say sa mga Cordillera Administrative Region, mga ganun. Dominant talaga. Sa NCR, doktora, we also don't notice, doktora, and that's why we are glad na yung ating NCR++ na ito, na-notice mo NCR++. Kasi doktora, when you really look at the sequences, yung mga iba-ibang mga variants na nandito sa Metro Manila, NCR, at saka mga variants sa Calabarzon, pareho sila. Nag-iikot-ikot sila. So, iba-iba sila. So, for example, marami tayong variants, doktora, may 40 lang, 40 different kinds of variants.
There are 40 different kinds of variants.
More. There are hundreds of variants around the world. Sa Pilipinas, so minsan isa lang ng ganito, kung isang marami. So, it depends, no? So, but at any rate, ang ating mga very common would be the Hong Kong and the UAE. At saka may Spanish variant din, doktora. Mayroon tayong Spanish variant early on. Yung B.1.1.74. Yun, marami sa Pilipinas din. No? May mga ganon, iba-iba.
Ma'am, narinig ko po kay former secretary, si Sir Manolet po. Sabi niya, and please tell me if this is correct, sometimes it depends po on the variant that's going around the country. Kaya kung minsan nahihirapan din mag-control kasi iba't iba po yung mga variants sa bawat
yeah. So, doktora, other than the variants of, marami naman, iba-iba. So, for example, if you sequence from Cagayan, Region 1, ang nakita ko, doktora, yung National Capital Region, talagang melting pot talaga tayo at ang Calabarzon. Yung National Capital Region at ang Calabarzon, syempre, it also reflects the mobility and the continuity of the borders, not really borders, contiguous borders natin. Makikita mo, doktora, na yung mga variants ng NCR at ng mga Kainta, ano pa ba yan, Binangonan, Calabarzon, pare-pareho sila. Samantalang if you go to other regions, let's say, get to Region 1, Region 2, merong mga predominant variants doon. Pero pag mag-NCR at Calabarzon ka na, makita mo different kinds of variants, makikita mo sa dalawang rehiyon. Kasi kino-color code namin nun sila, doktora, sa aming analysis. And that is really, really logical na mag-NCR++ tayo. Kasi ang ating nakikita sa genomic information ng virus is actually reflecting the mobility of people in the NCR and in the Calabarzon areas. Siyempre, doktora, doktora, di ba, kami sa Genome Center, nag-RT-PCR kami, di ba? Nagsisequence din kami. So, siyempre, marami kaming mga pasyente dito sa Quezon City, sa Cainta, Binangonan, Antipolo, kung minsan may Bulacan din, no? So, makikita namin, doktora, na kung ano yung pattern na nakikita natin sa Quezon City, maya-maya, yun na makikita natin sa Cainta. Makikita mo talaga siyang, nako, nandito na sa Cainta. Parang makikita mo, parang sa aming, alam mo sa early part, doktora, parang may 2 weeks delay lang. Ando na sa kabila. No? So, na-observe namin yan.
The sequencing, what will happen.
Oo. And we will notice also, doktora, na yung nakikita namin sa QC, nakikita na namin doon sa nearby municipalities of Rizal.
Opo. So, doktora, inuulan po tayo ng tanong. Would you mind to answer pero short lang?
Sure. I will answer short.
Para mapabigyan po natin yung mga viewers po natin. Opo. Paki-flash lang please yung mga tanong. Short lang tayo, doktora. And tell us
umiikot po ngayon na strains? Iba-iba, marami.
Yes, parang nasagot na ni Doktora yan
Mm.
that Hong Kong and UAE and ma'am, yung nakakatakot pong UK, Brazil and South Africa, umiikot na po ba or pa ano-ano pa lang?
So for example, doktora, kasi syempre nagsisequence kami in the beginning when we had to do a special run for Pasay. We did a special run for Pasay and the results came out. Wow, nakita ko may 351 kasi wala tayong 351 dati. Yung 351 is South African.
Ayun,
So yung 117. Ako personally, if you're going to ask me, syempre yung ano pa to? Yung sa nauna natin kasi doktora nakita yung 117, so we were very strict no with the contact tracing. Tapos yung nakita natin sa Pasay, so parang could this be the one that is driving the spike in Pasay? No, kasi for the longest time ang Pasay maganda ang kaniyang yung kaniyang record sa mababa daw sa Pasay, gano'n. Tapos we did another run for the National Capital Region with many more samples. Tapos lumabas ang dami na parang all of a sudden ang dami na natin ng 351, di ba? Marami ng 351. So, doktora, kasi it is in the nature of these variants of the virus to spread. That's number one. But ang ating mga variants of concern, 117 and the 351, for example, on top of the D614 gene mutation, meron pa silang 501 at saka 484 na that seems to be associated with increased transmissibility. So, sasabi naman din, doktora, ng WHO, itong mga variants of concern, hindi yung ordinary variants, yung mga variants of concern, variants of concern, hindi ito yung mga Hong Kong and UAE variants. Yung mga variants of concern, there are only 3. The P.1, we only saw it in the airport one, so I am not very concerned about that. But the B1117 and the B1351, which we seem to be found in almost... they are found in almost all cities of the entire NCR, no? Nakikita naman ninyo na nagsabi sa atin si DOH na naitalan na siya sa iba-ibang lugar ng Metro Manila. Sila kasi, doktora, and this is also for the information of our audience, itong mga variants of concern are highly infectious. Wala naman tayong talagang convincing data that they are causing severe symptoms, wala naman. They're highly infectious.
Pero mas nakakahawa lang po talaga.
Nakakahawa. So that is why, doktora, sa ganitong klaseng mga VOCs na ito, parang hindi umuubra ang home quarantine. Because everybody will get infected. And I think you observe that also na?
Government quarantine, yung ilabas po sa bahay yung mga tao.
Exactly. Kasi, and yan naman din sinabi ni Dr. Rabi ng WHO, kasi from the experience of the UK, Europe, and other countries, ang pattern niya, doktora, pamilya-pamilya ang nagpapositive sa mga VOCs.
Oo. The variants of concern. Sinabi ko na rin po yan kasi dati, isa o dalawa lang sa loob ng isang pamilya.
Agree.
Buong pamilya. So,
pag gano'n, doktora, So, syempre kung ganyan, di mapupuno din naman ang ating facility quarantines kasi there's also a limited number. But that's why it's very, very important na as much as possible, pag hindi necessary, don't go out of your houses. Do not go into crowds. I can also understand our human need, no, for contact, etc. But nang spread ito kasi we became complacent. Akala natin medyo bumaba na yung ating cases ng November, December. People became complacent. Kumain sa restaurant, sa tabi-tabi sa corner. Nagpunta-punta sa kahit saan. The virus did not go away. It's just here.
Ganun talaga. Opo. So, ma'am, I hope you will correct this information. Marami ako nakikita sa Facebook and paki-flash lang. There's a question about yung variants daw are caused by the vaccine. Parang sa akin,
that is, doktora, that is the pit. Doktora, that is the pit. And I think that is a very irresponsible, doktora, I think it's a very irresponsible behavior.
Yes, ma'am, talagang mali eh. Is there any
and it is not supported by evidence, and it's given a big audience. Nakita ko to. This is very irresponsible. And it undermines our efforts to increase vaccination.
Sabi ko bakit yung mga nabakunahan, hindi naman sila yung nagkasakit? Yung nagkasakit nga, yung mga hindi nabakunahan.
I
So, I
mean,
don't.
doktora, ganito, it's a personal opinion of a person. It is not vetted by scientific data or by referees, and it's given a wide distribution of audience. And I think it's grossly irresponsible, as in
Mali,
grossly
super mali.
irresponsible.
Opo.
So everybody, there is no proof to that, ha? And
Super mali.
magpabakuna po tayo because ang ating mga bakuna, even if, for example, there is a possibility of immune escape, makakatulong pa rin siya because it prevents hospitalization and of course the occurrence of severe cases. No?
Opo.
Naku,
Doktora,
doktora, ang dami talaga mga fake news at kung ano-ano. Anybody can post anything. Tapos some people with certain agenda. And then it's given a wide audience. So, kaya lumabas na rin ako, doktora, just to inform the people, to share from my point of view some of the things that we have been doing and that it is very important for the scientists and the medical professionals to communicate to our public the truth and for our public, huwag lang kayo makinig sa mga ganito. So, of
po,
course, siyempre, kung minsan ayaw ko naman siya bang super exposed, but sometimes we have to go out and to explain in a manner that is understandable na wala po scientific basis yun, ha? So, kung may opportunity tayo na magpa-vaccinate po tayo kasi yung ating mga vaccines, they have been vetted by the professionals, they have gone phase 3 clinical trials, and I think if you have the opportunity, get yourself vaccinated.
Thank you, Doktora. Doktora, napaka-inspiring po ng kwento ng Philippine Genome Center, and it's truly a Filipino accomplishment po. Lalo na for ladies pa ho. Can you, before we close, Doktora, thank you so much for your time. Can you give us words of encouragement for a young person who wants to enter the field and become a scientist po?
Oo, so I
Posible
think,
ho bang
yeah.
magka-career sa Pilipinas? Kasi na-touch din ho ako na sinabi niyo umikot po kayo ng mundo talking to various Filipinos who were in charge of labs. So parang
Correct.
what I get is that there is a big diaspora po of Filipino
Yeah,
science.
we benefited a lot from the talk of the different diaspora and also the foreign experts, no?
How can you inspire a young person na there is a career for him dito po sa Pilipinas?
If there is anything that came out of this pandemic was probably the fact that science is for the public good, no? So because of science, our scientists were able to develop a vaccine. Sabi natin parang maigsi lang ang pag-develop ng vaccine, but it's actually a journey of more than 10 years, more than 20 years, because the community has been waiting for a pandemic to happen. That's why it is not true that the vaccine was developed in less than a year. Not true, because the development process has been many years. That's one. And what I would like to say for the young people, I was really, really heartened, Doctora, when we started this pandemic. So many of our MBB volunteers, or many graduates, or MBB graduates, they volunteered to train our medtechs. Can you imagine? They volunteered so that we can have increased national capacity for RT-PCR. And siyempre, hindi naman lahat ng tao familiar doon sa RT-PCR and handling RNA, no? So we came up with that program called a CoLab. So this is with the University of the Philippines, in partnership with the DOH and the National Institutes of Health, MBB, UP Diliman, and the Philippine Genome Center, and also in Visayas and Mindanao. So people, scientists, and alumni reaching out to make a difference sa gawa natin. So if there is anything sa ating mga young people, I think science is a very worthy endeavor. It can be a force for public good, and for many people who are thinking and who believe that they have the aptitude and the talent for science, or even for computational biology and kung ano pang computational modeling. I think there is a career for science, and there's nothing more fulfilling for anyone to be able to help in this pandemic and for able to self-actualize what your talents are for public good. So I would like to encourage our young people, please pursue careers in science. It gives you, at the end of the day, it gives you a deep personal satisfaction that your talents are being used for the good of the public and to solve our country's problems.
Thank you, Doktora. Salamat po.
Oh,
So,
hindi na tayo nag-talk about cancer, doktora, no? Hindi na tayo
Right,
nakapag-usap.
part 2 na lang, ma'am. Part 2 po
Sige,
yan.
thank you very much, Dr. Anna, for this
Thank you,
opportunity,
ma'am.
no, to share our views and our
Ay, ako,
journey
I'm
at
so
the Philippine
excited
Genome
po. Laban
Center.
Pilipinos talaga tayo, ma'am. Salamat
So,
po.
Doktora, ha, in behalf of the entire Philippine Genome Center family, I would like to really thank the young people behind all these efforts and, of course, our collaborators from the Department of Health or the laboratories. It's a genomics network in the country and the many donors who have selflessly donated and made a difference really in this in our country's response for during this COVID pandemic. Thank you very much,
Salamat, ma'am.
everyone.
You're an
Stay
inspiration,
safe.
ma'am.
Yeah.
Yes. May nagsabi po dapat bigyan si ma'am ng medalya. One of my friends said upon hearing this that the Philippine Genome Center deserves a medal. Ako rin, I do believe. Kasi biro nyo po, if we didn't have this, we don't even know kung anong variant ang umiikot sa Pilipinas. These people are heroes. Sila po ay COVID heroes. It's really so nakakataba po ng puso. Marami pa hong tanong eh. Pwede po tayo umabot ng 3 hours, kaya lang nakakahiya na po. Dr. Cynthia Palmes Saloma, and the young persons po from the Philippine Genome Center, you are an inspiration po. Nung nagsari-sarili na po ang bawat bansa, kasi hindi na po pwede na makiki-sequence ka sa ibang bansa. Siyempre, sini-sequence nila yung kanila. They were able to do this. So, Ms. Rains, can you send a few questions and I'll just try to answer all of them. Thank you so much. Sorry talaga, daming sinabi ni ma'am, pero lahat magaganda.
I agree, Doctora. Okay, so, Ms. Kat, ready na po ba with the questions?
How do the variants arise? Ma'am Cathy, ganito po 'yon, ano. As the pandemic progresses, dumadami po yung virus kasi ang daming nagkakasakit, ano po. As the virus moves from person to person, and as the virus just multiplies po in our body, nag-iiba po yung genetic code. Kung minsan nagkakamali, there just are mistakes in the genetic code. And then, these mistakes po are called mutations. Ngayon, kung yung mutation po, there are bad mistakes eh, like humihina po. It's like ano yan, nanganganak. Sa pagpapanganak mo, sometimes makakapanganak ka ng mas mahina. Yun ang namamatay na mutation. Kung minsan sa panganganak mo, nakakapagpanganak ka ng mas malakas. At itong mas malakas na to, ito ngayon yung papangibabaw. Kasi para kang nanganak, Rina, para kang asong nanganak ng 11, okay? Asong nanganak, let's just put it na may aso ka, nanganak ng 11. Yung mga 11 na yon, mga anak mo yon, some of them will have different genetics, okay? Yung mahihinang aso, mamamatay. Yung mga malalakas na aso, siya yung papangibabaw at yun ang mga anak ng marami. So yun ang mutations. Na-gets nyo yun. And in viruses, it happens. Viruses don't need a male and a female. They by themselves nanganganak sila. Sa pagpapanganak nila, naiiba yung genetic code nila. Yung mahihina, namamatay. Yung mga malalakas, they persist at yun ang magkakaroon ng marami pang anak. And these are what we call mutations. Okay? Mutations. So, pag mas nakakahawa, which is the variants of concern, ito yung nakuha ko kay doktora, there are millions of variants. But there are only a few that are what we call VOC or variants of concern. At yun yung maraming anak. And mas variants of concern sila dahil yun ang mas nakakahawa. Ito po yung tatlo, yung UK, yung South Africa, at yung Brazil. Whether or not the P.3 will become a variant of concern, hindi pa po natin alam. Nakuha mo ba yon, Reyna?
Me, yes, Doctora, na-gets ko po. Actually,
kasi, Dr. Saloma is an excellent scientist. Kaya lang, there's a difference also between being a scientist at yung naiintindihan sa barangay. Kung minsan mahirap.
That's true, Doctora. And I think malinaw naman yung pagkaka-explain nyo po na yun nga yung mga variants, nanganganak talaga. So, opo.
Pagpapanganak nila, merong mahinang anak at merong malakas na anak. Yung mahina, mamamatay. Yung malakas, yun ang mangingibabaw at yun ang magkakaroon ng maraming anak. So, yun yung mga variants.
So does it mean, Doktora, say for example, Brazil, Philippine, South Africa variant, ibig sabihin ito yung, kaya sila tinawag na Brazil variant, South Africa
So, doon
variant.
sila na-discover.
Okay, that's clear.
Doon
That's
sila
very
na-discover. Pero again, sabi nga ni Doktora, and I would like to say it's P.3, no? Kasi, ikaw ba, pupunta ka pa ba ng UK ngayon? O pag may narinig kang galing ng UK, baka susi mo yung pintuan mo. What I'm trying to say is may discrimination talaga. So I'm
yes.
hoping that that won't happen to the Philippines. Kasi syempre, ang dami nating overseas workers, dami nating mga products, ganon. Pag
Mm.
napagsagan siyang Philippines, maraming matatakot. Ikaw ba, maghaharap ka na sa taong tiga UK, Reyna? Baka
Ako, hindi.
yung pintuan mo.
So another follow-up question to that question of mine, Doktora. So would that mean na halimbawa since UK variant na discover siya sa UK, does it mean ba na pwedeng magkaroon ng UK variant dito sa Pilipinas without
Meron na.
without an exposure to a UK
Ganito
carrier?
yun. Okay, ganito. Okay, I got your question. Pwede ka
Yeah.
bang magkaroon ng UK variant without exposure to someone who came from the UK? Iyan ang tinatawag nating community transmission. Tama ba ako sa tanong mo?
Yes, you got it correctly,
Okay,
Doctor.
ganito. Okay, assuming, and I don't, let's say there was a traveler from the UK. I don't want to say OFW kasi discriminatory na naman yon, di ba? So let's just say there was a traveler who had the UK virus, okay? And then came to the Philippines, okay? Wala pa siyang nararamdaman. Tapos, let's say, kasi di ba remember dati, isang araw lang yung quarantine sa airport, then labas ka na. Remember when we talked to Ma'am Syra at saka yung pamangkin ko? Naka-isang oras, overnight lang sila sa quarantine hotel, negative, labas. Alam naman natin na yung RT-PCR, hindi naman 100%. Kasi for example, nandun ka it depends kasi kung anong araw, usually 5th day. Halimbawa, the day before or sa airport, doon ka naubuhan ng UK virus. Pagdating mo sa Pilipinas, day 2 pa lang yun. O, di negative si PCR, di lumabas ka. O, sige lumabas ka. O ngayon, ikaw, pumunta ka sa family reunion. May nagkasakit doon ng mga kamag-anak mo kasi kumain ka na walang mask. Okay. So, ang dala mo yung UK variant. So, yung mahahawa sa iyo ngayon, let's say yung yung isang waiter doon or pamangkin mo or whatever, nahawa ng UK variant. So, ikaw, after 5 days, nagkalagnat ka na, pumunta ka na sa ospital. So, kinontact tracing lahat ng nasa party. O, let's say yung pamangkin mo, kaharap mo, kumain kayo, nagkahawaan kayong dalawa. Okay, si pamangkin now works somewhere else. Kumain naman ng lunch si pamangkin dun sa kanyang opisina. O, nagkaharap sila ngayon with the office mates. Si office mates, si pamangkin, hindi nanggaling ng UK. So the office mates can get the UK virus from the pamangkin who was exposed to the person na ikaw na galing sa UK. O, so si office mates, that is what we call community transmission. Kung yung pamangkin mo nakipag-inuman at kainan with the office mates, kumakalat na siya from the original, which is the person who came from the UK mismo. And yan ang ayaw na ayaw nating mangyari, which is community transmission. Kasi pag hindi mo na natitrace kung saan na nanggaling, ayun na yung community transmission. Sasabihin ng mga office mates, wala naman ho kami nakilalang galing ng UK, paano kami nagkaroon ng UK virus? Ang problema, kumain sila dun sa isang taong nagkaroon ng exposure dun sa galing sa UK. Kung yung mga office mates na to, nalima sila, kumain pa sa bahay nila, mas lalong na-gets mo, Reyna? Dumadami ng dumadami.
Yes, Dora. Opo, nag-gets ko.
So the office mates are like, wala naman kaming kilalang taga UK, paano nangyari to? Oh. Pero, yung ka-office nila, yun ang nakipag-family reunion dun sa galing sa UK. Paano yun? Yun. In practical terms, Tapos, yung office mates, kumain dun sa bahay nila, lahat ng nasa bahay na infect. Ayan na. That is what we call community transmission. Yung hindi mo na malaman Bakit ako nagka-UK virus? Di naman ako galing sa UK at wala akong kilala from UK. Kumakalat na sa community at yun ang community transmission at yun ang nakakatakot talaga. Super.
Oh no. Oh
Oh
no
no.
talaga.
Oh no, talagang oh no.
Mhm.
Can you get COVID variants if you have had COVID? Ma'am Cathy, ito ang nakakatakot sa Brazil variant because there are reports coming out of Brazil and that is out of the city of Manaus, 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 Brazil. Kasi sa Manaus, Brazil, nagkaroon na sila ng first COVID wave. So ngayon, may COVID wave na naman sila. Nagkakasakit na naman yung nakasakit na nung first COVID wave. So there is a fear that the virus can bypass, the variant can bypass known yung defenses na antibodies at memory and T and B cells. So yon nakakatakot rin. So
From
Doris.
Doris Guerra Cunanan, ano po bang asymptomatic COVID? May virus po ba sa body? Bakit walang nararamdaman pero pag nagpa-test is positive? Posibleng mga nakasalamuha na hawa na rin. Salamat po.
Yes, ma'am. Ito po ang worry natin po sa asymptomatic. Sometimes hindi naman po talaga asymptomatic eh. There are two terms. Pwede kong asymptomatic. Asymptomatic, yung talagang walang mararamdaman pero nagkakalat. Meron din ho nun, but it's not that big. Okay? Meron din ho, ito yung mas nakakatakot, yung tinatawag po nating pre-symptomatic. Ano po yung meaning nun? What is the meaning of pre-symptomatic? The meaning po of pre-symptomatic is, may COVID na siya, pero wala pa siyang nararamdaman. So, umiikot-ikot siya ngayon. At umuubo-ubo, kumakanta-kanta, kumakain-kain, nagkakalat na po. Okay, how is this happening? Okay, balik tayo. Day 1, day 0, kumain ka kasama nung isang galing na nga ng UK. Sige, sabihin na natin UK. Huwag daw mag-discriminate. Nagdi-discriminate tayo rito. Okay, so let's say day 0, kumain kayo with your relatives who came from wherever. Okay? Tapos, ikaw, nagtanggal ka ng mask, kumain ka sa harap ng mga tao, nakipagkwentuhan ka pa. So, nahawaan ka ngayon. Okay? After, since wala ka naman nararamdaman, umiikot-ikot ka. Okay. Day 4, may tumawag sa'yo. Meron hong nag-positive dun sa party, mabuti ho magpa-test kayo. Ikaw naman is like, wala akong nararamdaman. Meanwhile, pumunta ka na sa trabaho, nakipag-inuman ka sa kumpari mo, nag-mask ka at kumanta-kanta ka pa doon. And on day 4, nandun ka sa, umiikot walang mask. So, on day 5, bigla kang nilagnat. Oh my gosh. Tapos, day 6, nagpa-test ka positive. So, nag-quarantine ka na. Ay. Pero ang tanong, anong nangyari between day 1 to 5? Yun ang tinatawag na pre-symptomatic. Wala ka pang nararamdaman, pero nahawaan ka na. At sa pagkahawa mong yun, nagkakalat ka na po. Yes, nakakahawa yun. Nagets mo ba, Reyna?
Yes, doktora. Napakaganda
Yes,
ng...
nahawaan ka on day 0. From day 0 to day 5, wala kang nararamdaman. Day 5, nilagnat ka, nagka-test ka, nagpa-quarantine ka, day 6. Anong nangyari between day 1 to 5? Oh. Anong ginawa mo? I hope you followed the COVID rules. Kung nakipag-inuman ka, kung nakipag-party ka, kung kumain kang walang mask sa harap ng maraming tao, patay. Nagets?
Yes, doktora.
So there are asymptomatic, Ma'am Doris. Yun yung mga taong wala nang mararamdaman, gagaling na lang ng COVID. Merong ganon. Pero majority po talaga ng mga tao is what we call pre-symptomatic. Wala pa lang nararamdaman, pero nahawaan na po at nagkakalat. Okay? At yun ang nakakatakot. So whether or not, alam mong may COVID o walang COVID yung isang tao, mag-mask kayo. Period, wala nang tanungan. From Tony.
Here. From Mr. Tony G. Doc, paano po yung lumalagpas sa criteria of beyond 30 of cycle threshold? Kapag sumobra ng 40 to 45 CT, ano po ang epekto nito sa result?
Tony, I'm not a microbiologist, pero what I understand is this, okay? And please correct me if I'm wrong. The higher the cycle threshold, that means mas mahirap makita yung COVID. So it's possible na pag masyado na pong mataas yung cycle threshold, mga remnants na lang po yun. Kung mababa yung cycle threshold, it means marami ka talagang COVID at madali siyang makuha. So the higher the cycle threshold, the likelier na may remnants or patay yung COVID na yun. Please correct me if I'm wrong because I'm not a microbiologist. Okay, babalikan ko yung tanong na yan.
From Miss Kathy Rodriguez, do the new variants have different symptoms?
Ma'am Cathy, mukhang pareho pa lang yung mga symptoms. Kaya lang, mas nakakahawa siya. Kasi dati, ito, and I would like to, na-mention ko na rin po ito. Dati, kasi kami, contact tracer kami. Dati, pag merong may COVID sa isang bahay, di ba, syempre, magkamag-anak yun, hindi sila kakain na nakamas. Isa o dalawa ang mahahawa. Pero ngayon, dahil mas nakakahawa, nakikita po natin yung buong bahay. From the kasambahay, to the lola, to the kapatid, to the driver, to the labandera, everybody gets COVID. So yun po yung yung pagkahawa niya. So that's really worrisome. And then number 2 po, sa UK po, it's not only more nakakahawa, but I read a study saying that it is 55% more nakakamatay. So it's deadlier yung UK. And then number 3, ito lang. For example, kasi 2% talaga yung death rate ni 2% ang mamamatay and 20% ang ma-hospital. Okay, so marami nagsasabi, ba't tayo mag-worry? 2% hindi naman dumadami yung mamamatay. Okay, ito lang. Ito, kung 10,000 lang ho yung may COVID, so what is 2% of 10,000? Mababa lang po, and 20% mababa. Kung 100,000 na po yung may positive, di yung 2% na yon mas malaki po. So, mas marami yung mamamatay automatic just dahil mas marami. At saka, nakakatakot po ito, yung pagsiksikan po ng ospital ng 20%. Kasi magsisiksikan yun sa ospital, hindi naman dumadami yung ospital ng Pilipinas. So, yun po yung masama kasi the hospitals will become so crowded kasi sobrang dami because mas marami silang may sakit. Okay? So ngayon, pag nakipagsiksikan lahat ng tao ng COVID sa ospital, so mas marami pong mamamatay because 2% of 1 million is bigger than 2% of 10,000, obvious ba? And the bad thing is, paano yung naatake? Paano yung may cancer? Paano yung may pulmonya? Hindi na, paano yung may dengue? Hindi na sila pwedeng pumunta sa ospital dahil na-take up na ng COVID lahat. So lahat yon mamamatay rin. So basically, yun po yung 2 reasons kung bakit mas nakakatakot po yung variants. Number 1, 2% of 10,000 is definitely less than 2% of 1 million. And the 20% po na makikipagsiksikan sa ospital, mapupuno po yung mga ER. And then, hindi lang po yung mga COVID, syempre, yung may na-atake, yung nasagasaan, yung nakapagsaksakan, yung nagbarilan, yung may cancer, yung may dengue, yung may pulmonya, hindi na sila pwedeng pumunta sa ER dahil nandun na lahat ng COVID. Nakuha mo yon, Reyna?
Yeah, Doktora, and you know what makes everything so sad? Yung mga mababasa mo sa mga comments and other stuff na naku, pinapataas na lang naman nila yung number. Totoo ba yung number na yan? O pinapataas? That's so sad. That is so sad. You go to each and every hospital. Ikaw, Doktora, nag-ikot ka ng mga hospital.
Si husband ko, nasa St. Luke's ER nung isang araw. Wala na pong maupuan yung mga pasyente, tabi-tabi na lang. Okay? COVID and non-COVID, basta nakamask. Akala nga ng husband ko, yung mga COVID, lalagay dito yung mga non-COVID. Hindi po, pinagsama na lang at bahala na kayo mag-mask. Okay? Ganun na. So it's really not, kasi the waiting room talagang grabe na. Okay? Number 2, sa Cardinal. 3 days ago, may pinila po kami, number 71. 2 araw na ho, number 31 pa lang siya ngayon. Okay? So don't think, paano may cancer? Wala na. Imagine, it took 3 days, wala pa kung And kung hindi ka pupunta doon, sa araw na pupuntahan ka sa ER, ang dami-daming gustong kumuha ng pwesto mo. So yun lang. If you don't believe COVID, you don't believe it's real, please tumulong na lang po kayo sa ospital. Gusto nyo maglinis kayo doon or what, i-volunteer nyo yung sarili nyo dahil talagang pagod na rin po yung mga frontliners sa ospital. Okay? Next.
From Mr. Ricky Lee Mangaliyag. Airborne po ba ang mga variants kaya madaling mahawa ngayon?
Yes,
Thank you.
all COVID, variant and non-variant is airborne. Nakakahawa po siya because there are modifications or mutations po sa kanyang spike protein that allows them to enter human cells faster.
From Miss Santa Rosalia, how do you know that these variants are African, Singaporean, British, etc.? I am not a lab chemist, but most of us listeners do not know these terms.
How do we know? May passport po sila. Hindi, biro lang. Ma'am, thank you for this question and I'm glad that you asked this because marami po talagang nagtatanong. Ma'am, usually, they are named po after the place where they were discovered. Okay? And tama po si Ma'am, si Doktora Cynthia na discriminatory po talaga kasi ikaw ba lalapit ka sa South African ngayon? Ako talagang ayoko pati yung mga products nila hindi ko hahawakan. Kahit dumating pa si Prince William dito na mahal ko yon at si Prince Harry, baka hindi ako makalapit. So basically it's discrimination. Kaya lang, I would agree rin with ma'am na dapat merong mga proper naming kasi ang hirap po yung B1117, B111.8. Opo. So thank you ma'am for this question and ang hirap-hirap po talaga na ma-explain po ang science kasi science is not easy. Mahirap pong ma-explain ang science sa mga normal na tao. Ay ako nga doktor nahihirapan na ako eh. Kailangan ko nga sabihin kay ma'am, slow down lang, backtrack. Tama ho ba yung pagkaintindi ko? But we have to do this because if people do not understand science, doon po lumalabas yung mga wrong science. Alam mo yon? And doon na ho lumalabas yung mga kung ano-ano. So we have to understand or seek to explain po kung ano yung tamang nangyayari para maintindihan po ng mga tao para po mag-cooperation sila. Kasi pag hindi nila naintindihan, hindi sila mag-cooperate dahil hindi nila mag-gets. Nakuha mo ba yun, Reyna?
Yes po, Doktora.
Oo, kailangan mag-gets ng tao. So, thank you for that question. I just made a joke para masyado na talagang sad. Parang lang ngumiti lang tayo ngayon. Opo. Ayan, from Ma'am Santa. Sige, pakibasa lang po.
From Miss Santa Rosalia, thank you so much, Dr. Ana, for understanding us laymen, for supporting us in explaining and making Dr. Saloma also in making her explanation simple to us. Thank you so much, Dr. Ana, for helping us in these terms you do understand as laymen.
Ma'am, thank you so much. Yun po talaga yung plano ko talaga sa buhay. Alam kung bakit? Kasi I've spent a really long time as a doctor and I've really spent a long time then po. And you know, sometimes politicians are unfairly maligned. Kaya lang, yung kakwentuhan ko rin po with much respect. I have the same respect for a barangay tanod and for a barangay health worker, the same respect po na binibigay ko po sa isang governor at sa isa pong doktor sa isang malaking ospital. Pareho lang, okay? And pag hindi po natin naintindihan, pag hindi po natin mapaintindi sa bawat Pilipino, it's a national emergency, it's a global emergency. If we cannot explain to them what is happening, how can they help us? Alam mo yon, we cannot command people na, hoy, magpabakuna ka. Or, hoy, doon ka sa bahay mo. Hindi pwedeng ganun eh. Kailangan i-explica natin bakit po kailangan magpabakuna. Bakit po nandun po tayo sa bahay natin. This is not a dictatorship. And we have to get the cooperation and understanding of everyone. Ako, doktor na rin ako. Matagal na rin yung ko. Kung minsan nagugulat din ako. Like when my manager asked me the question, Doktora, what is asymptomatic? Akala ko automatic na yun. And that person was knowledgeable, hardworking, a regular Filipino. So basically, yung akala po nating alam ng tao, hello, hindi nila alam. And it is up to us to explain it to them. Otherwise, wala tayong karapatan na magalit kung ayaw nila magpabakuna o ayaw nila mag-stay sa bahay nila. Yun lang.
Yeah. So from Miss Santa Rosalia again. Dr. Anna, you have my heart and you have helped me in this few minutes talk regarding medical terms, which could be foreign to most of us. Grateful to you, Dr. Anna, and thank you to Dr. Saloma. Dr. Anna, I am a follower of yours in your rural and urban activities.
Thanks, ma'am. And if you don't understand, just ask. We'll try our best again. Opo.
Please flash Miss Kat. From Miss Mavic Kiambao Pama, salamat po sa napakalinaw na explanation about community transmission.
Thanks, Mavic. Mavic, salamat. I'll PM you later. May pasalamat din ako sa iyo personal.
From Miss Remy Reyes, good day, Doctora. In what level do Philippine variants dominate now?
Ang pagkakuha ko lang po kay mama, ito lang. Parang may na ano ko sa kanya. I was able to write down something. Sabi niya po na may nasulat ako how many variants na nasa Pilipinas. I think 100 plus, there are 104 na raw na na-sequence nila. Pero again, Rami, ito lang I got also from Dr. Saloma. They do not sequence every positive test. And ang maganda ito, Rina, the PGC, the Philippine Genome Center, cannot sequence all the positive tests. They try to get representative samples from every positive test that happens. And then, number 2, pinapasok din po nila yung mga lugar na bigla pong may sipa paakyat. Like, napansin po nila sa Central Visayas, may sipa. So, they rush there and nalaman nila may Philippines variant. And then, sa NCR, may sipa pong paakyat bigla from 1,000, biglang nagiging 5,000. They are rapidly sequencing din po sa Metro Manila to understand the situation on the ground. Ang ganda nung sinabi niyan yan, and that's something very important.
So, yes, kung nagtatanong po ang mga karamihan sa tao, kung meron pa ditong, kung nandito na ba ang mga variants, nandito. Na po.
Nandito. Sinabi niya po, it is located all over Metro Manila. And what talagang kailangan may gawin po ang ating gobyerno regarding this. Aside from the government, you yourself have to do your part. Yun lang po.
Yes. Another one, Miss Kat? From Miss Remy Reyes, very clear explanations po, Doktora, regarding local transmission. Thanks po for
your... Naging clear kasi talagang sa barangay po ako nag-i-explain talaga. So, talagang ano po. Tsaka ano, yung mga kinakausap ko, ito lang kung minsan mas malinaw pang kausap ang mga barangay tanod kaysa mga doktor. Sorry, mga doktor. But people who live, ito lang people who live in the province or are not necessarily doctors, it does not mean na mas less ang knowledge nila. Some of them, most of them are very practical. They have to work every day. They know what to do. They are seeking information. So just because someone lives in the province or someone is not a doctor doesn't mean na hindi niya alam. It is our job to communicate science properly to everyone. And remember, everybody is an influencer po kasi everybody has a family. Sa pamilya nyo na lang po yung sinasabi at ginagawa nyo, malaking bagay na po yun.
Okay.
Kung kayo po ay nagkakalat ng mali sa kamag-anakan nyo, patay. Pero kung kayo po ay nagkakalat po ng tamang impormasyon, malaking tulong na po yun sa buong Pilipinas. Everyone is a hero po and everyone's opinion matters, lalo na po sa pamilya nyo. Kung kayo po ang kuya, yung influence nyo na lang po sa mga kapatid, malaking bagay na yun.
Okay, Doctora, we have a comment. Miss Kat? From Miss Angela Yang, why don't you help the people inside the Malacanang to what is happening now? They don't know anything about the pandemic virus. They are not specialists who studied the technology of virus. Why don't you talk to the president, which is good for us?
Thanks for this comment, Ma'am Angela. And feeling ko na frustrate rin po kayo sa nangyayari. And I would like to say, everybody is frustrated and sad. And I would like to also say, this pandemic is not happening lang po sa Pilipinas. Right now, merong headline po. Germany, kumakalat po ang UK virus sa Germany ngayon, and Angela Merkel just called it a new pandemic. Okay? So siguro we should also educate Angela Merkel and Boris Johnson, and somebody educate Donald Trump. So what I'm trying to say is, I understand po yung frustration. I personally am frustrated. May business po akong pabagsak na. O what? But what can we do? What we can do po is, I understand the need to blame the government, the need to blame everyone. And I'm not defending the government. Marami rin pong pagkukulang. But I think that we are dealing with a difficult enemy, one which is worldwide and which is happening po throughout the world. Ngayon po, but that doesn't excuse rin po yung pagkakamali ng gobyerno. Because meron din pong magkakamali. We can ask the following questions. Bakit hindi tayo naging mas strict sa airport? Bakit po hindi alam mo ba sa Hong Kong, 21 days, no questions asked, wag ka na magtanong. You cannot enter Hong Kong. Oh, ako, gusto ko yun. So what I'm trying to say is, mahirap naman hong sabihin, I think the people in Malacanang are knowledgeable. They also po have advisors. Nakikinig din po sila. And they are trying to craft a delicate balance between our economy and our safety. So, naiintindihan ko po yung frustration po talaga nating lahat. It's frustrating. It's sad. But ultimately po, kaya nga siya pandemic, it's happening all over the world. But again, that doesn't stop us from seeking to do better every day. So the government must improve themselves, and lahat tayo, we must improve ourselves also. May nagsasabi, no, ang dali namang magsabing stay at home, may kinikita kayo. Paano po yung kailangan magtrabaho? I totally agree with that. I have much sympathy for those na arawan. Kawawa po sila, and really, we must support them. Kaya lang, parang wala naman ho sa essentials yung mag-party, mag-inuman, kumain sa mga kapitbahay at mga family reunion na walang mask. Parang wala naman ho yun doon. Okay, so doon po nawala na ako ng sympathy.
I think what you are trying to also say to us, Doktora, is gumulong pa rin ang ekonomiya, pero let's be responsible. Ano, Doktora? Yung simpleng...
Oo, meron din.
Yung simpleng pag-advise nyo lang po na huwag naman kumain ng sabay-sabay. Number 2, lalabas ka ng bahay mo, wala kang mask. Kahit ba tumambay ka sa harapan ng bahay mo, mag-mask ka pa din. Paano kung may dumaan na nagmomotor, umubo? Diba? So, yun yung mga bagay na
first government responsibility, at hindi ko po sila ine-excuse na marami ring mali. Okay? But there's also personal responsibility. So, sana ayusin natin yung dalawang yun.
Agree. Okay. Meron pa po ba, Miss Kat? Because we are nearly 2 hours from Mr. Ignacio Lim. They should have hired more doctors who is trained to save lives instead of a military man.
Ito lang madaling magsalita ng ganun eh. They should have hired doctors. But you know what? Asawa ako ng mayor, I know what's happening on the ground. And, you know, doctors, at saka yung IATF, nagbibigay lang po sila ng rekomendasyon. Alam mo ba kung sino nagpapatupad nun, Reyna? Mayors and barangay captains. So, I think what we need is a multi-sectoral group na iba't iba po ang kontribusyon at kaalaman, at magtulungan po tayo para matapos na po itong pandemic na to. Hiring all doctors I work with all doctors every day. Hindi po nila alam talaga kung ano nangyayari sa barangay. Pababain mo yan, hindi man nila alam kung ano yung trabaho ng barangay healthcare worker. Basically, what we need, and I saw that from Dr. Salima, she's a research scientist. She's working with epidemiologists. Ngayon po, what they told us, we have to apply it as the barangay captain, as the mayor, as the doctor. So, kailangan po talaga multi-sectoral. Kasi may naglagay, palitan yung IATF, palitan. Okay, sige, palitan natin. After that, what's the next step? Wala na hong nakasagot eh. So, you know what, madali lang magpalit ng tao. What we need is is a cooperation between various groups. If there is anything that COVID has told me, it's that no man is an island. Okay, kahit na ikaw na po ay bilyonaryo, you still have to go out and make palengke. And your business still has to exist, and you have workers. So basically, no man talaga is an island, and we are not going to finish this problem kung magsari-sarili po tayo. We will be able to solve this problem if we all cooperate po together and magtulungan. The government with the proper policies, mayors and barangay captains with enforcement of that policy. Hindi po si President Duterte ang nagka-quarantine sa bahay, okay? Hindi po siya yon. Ngayon, sa mga barangay captain, please, and I cannot understand this na sinabi rin, ba't ba hindi ilabas yung mga tao sa government facility? Bakit po sila nagka-quarantine sa bahay? I cannot understand this. Yun po talaga yung kailangan talaga eh. Ako, I'm in favor of ilabas po sa bahay yung mga positive.
Agree, kasi doon din
Yun po
naman.
yung ginagawa namin sa province. Muntik ko na nga akong tinapon yung asawa ko sa quarantine facility nung umubo. But luckily, hindi naman pala. So basically, everybody, my point in this, everybody has a role to play sa pandemic. We have a right rin po to demand po from the government the proper services. So yun po yun. Pero hindi po ang gobyerno ang pipigil sa iyo pumunta sa party at makipaglamunan at inuman doon. Okay? So tingnan nyo rin po yung responsibilidad nyo at yung ginawa nyo para ho nangyari to. Nagbakasyon ba kayo sa kung saan-saan? Nakipag-inuman ho ba kayo? Okay? Kasi yung pumunta ka sa trabaho, I totally understand that. Pero iba na ho yung gumala sa daan na walang mask. Iba na po yun. And I don't think that is the fault of President Duterte any longer.
Agree ako, doktora. Tsaka yung halimbawa ba na kung hindi ka naman naniniwala na may COVID,
Naku,
pwede
isa pa yan.
ikaw na lang yung pumasok sa loob ng bahay mo, huwag ka na lang lumabas. Kasi baka mama ikaw pa yung carrier. And please be respectful of others who believe and namatayan ng COVID. Please be respectful on them. Wag nang, ay hindi ako magmamask kasi naman ako naniniwala sa COVID. Bukod doon, nagkalat ka pa ng chismis, di ba? So, please respect. We need also some respect, yung mga naniniwala. Di ba, Doc?
Ako po, I don't want to say of personal stuff, but today is the first year anniversary po of two of my cousins. So yun na lang. Linagay ko po dun sa family Viber group, ang pinakamasakit pong nangyari sa akin, yung hindi ako nakiramay at hindi ko po napakita sa mga tao yung love and concern ko po sa kanila. So, I have no words for these people, Reina. Let's just move on.
Okay po. Well, on a positive side from Miss Cathy Rodriguez, salute to Dr. Anna for being passionate and patient to explain it easier for all of us. God bless us, everyone.
Thanks, Ma'am Cathy. Talagang ano po we cannot expect people to move if they do not understand why.
Agree. So that's all for today, Doctora.
Thank you. I'd like to thank Dr. Cynthia Palmes Saloma. Ma'am, we are really grateful kasi alam po namin na ang dami daw nag-interview sa kanya pero she wanted to talk to us kasi yung approach natin was scientific. And which I truly agree because I'm a scientist myself. I went into the sciences. I'm actually interested very much po dito. Though ang ano ko rin, because of I'm a politician and a psychologist, napunta po ako sa social sciences. But sometimes it's good to have people who are bridge between two very important aspects of our society. Yung science po at saka yung mga tao kasi kailangan mag-meeting din po sila. So ma'am and the Philippine Genomic Center, you are COVID heroes po talaga. And ang ganda-ganda ng kwento na 4 na babae pala ang nag-start nito, Reyna. Grabe. Okay?
I didn't know that. That's how powerful
4 na
everyone.
Maria daw sila. And they started this. And when the pandemic struck, we were ready. We had the equipments. We were able to sequence our own COVID genomes. May part 2 pa. We'll have a part 2 of this later during the year when mom is not so busy anymore. And pag sana natapos na tong search na to, my God. Para ho, I'm very curious about what the Philippine Genome Center was doing pre-pandemic, how they help the industry and persons, how we personally can benefit from what they're doing. Meron po silang trabaho about cancer, meron po silang trabaho about the Philippine genome. They are planning to sequence the Philippine genome. So ang dami pong magandang usapin dito. Napakahaba lang po talaga. And but at least it gives us a part 2, 'di ba? Para malaman natin. Okay? And for young persons who would like to go into science, the Philippines is here, the Philippines is ready. And yes, you can have a good career po. A good and a productive scientific career dito po sa Pilipinas. Salamat po sa inyong lahat. And mabuhay po ang Philippine Genome Center. Thank you so much.